Morning, everyone. I'm Tintin Su. Welcome to the MCO retreat. I'm one of the co-leaders here. And I'll just say very briefly that the way we're going to do Q&A is if you have a question that can wait till the end of the talk, just put your name, not the question, just your name in the chat, and we'll be calling you in the order that they appear. If there's something urgent that you want to ask, of course, you can use the raise hand function during the talk. Okay. And we'll be monitoring all of that. So with that, I'm going to hand it over to James, Deputy Director of the Cancer Center, to introduce our keynote speaker. Go ahead, James.
Well, first off, thanks Tintin, Craig, and Patricia for organizing this. It just is a great schedule. And it's really my pleasure to introduce Julie Cooper. So Julie Cooper keeps trying to get away from BMG, but we just keep drawing her back. So for those that don't know it,
yay!
So Julie did her PhD in Department of, it wasn't biochemistry molecular genetics then, but it essentially was this department back a while ago. And then she went off and did postdocs with people that either had Nobel Prizes or were about to get them. So she did a postdoc with Tom Check in Boulder and a postdoc with Paul Nurse at the ICRF in London. And then as the prodigal daughter, she got drawn back to the department as a junior faculty member back in 1997, and for the same search that I actually joined the department. And then a few years later, she got attracted off again back to, I believe it was still the ICRF, right, Julie?
Yeah.
As a faculty member there, and spent a good while there doing fantastic research. Then she came back to the States, but this time to the National Institutes of Aging as part of the NIH, where she has been up until last spring, or this very close spring, where she came and took over at the chair position for our department. And we're just delighted that she's our chair. Her work is really, I think the wonderful thing about it is she's absolutely addressing fundamentals, how telomeres function. But I find some of the most interesting stuff, and I'm not sure if she's going to talk about it, is how cells manage without telomeres. And this is particularly important in cancer because there's a fraction of cancers that don't reactivate telomerase. And basically they have to come up with alternative mechanisms. And she's shown that there are these repeat sequences that get used at the end of that get chromatinized at the end of these telomeres that can essentially substitute for these telomeres. So we're really delighted that she's back in Colorado once again, and that she is chairing us, and really look forward to her talk. So take it away, Julie.
Thank you, James, for that really nice intro. Certainly one of the great pleasures of being back here has been able to interact again with James. And I really I was hoping that I'd be able to see you all on the day of the retreat, I mean in person, and hopefully that will come. Okay, so I'm gonna start sharing my screen, hopefully. Okay, is that happening?
Yes, it all looks great.
Okay. And just to say, I'm a little incoherent in the morning, so really if you want to interrupt me for clarity or whatever, go for it. So I'm going to talk to you today about telomeres and centromeres, actually. So these are the two most prominent landmarks of the chromosome, and maybe the most important in terms of protecting the stability, the integrity of the genome. Centromeres, of course, are the structures that connect the chromosomes to the spindle so that they are distributed perfectly accurately to new daughter cells at every cell division. And the telomeres I'm going to talk about momentarily. So I'm going to start by introducing telomeres and some of what we know about the relationships between telomeres and tumorigenesis in cancer. And then I'm going to tell a new story from my lab that is kind of a surprise about a way in which telomeres actually quite directly affect centromere function. And some of the kind of highlights of the story are the importance of location of these regions in the nucleus, and just how studying meiosis and fission yeast can lead to insights about unforeseen cancer mechanisms. So it's going to start with meiosis and fission yeast and end up in human cancer cells. Okay, so the telomeres are, of course, the ends of the chromosomes, and they consist of a number of things, the DNA and the proteins that bind. I'm just going to, can I get myself into speaker mode or something so I can see my slides? Yeah, sorry. Okay, it's good. So the terminal sequences of all vertebrate chromosomes consist of TTAG3 repeats. So in human cells, there are about 10 to 15 kilobases of those, terminating in a 3' single-stranded overhang of the G-rich strand of the telomere sequence. There are proteins that bind the double-strand region of the telomere and the single-strand overhang, so telomere-specific DNA binding proteins that interact with further proteins to make the telomere complexes. And they do a couple of very classically recognized fundamental things. They prevent chromosome ends, the natural ends of the chromosomes, from triggering DNA damage responses. So unlike what happens when you have a damage-induced break in the middle of the chromosome, our natural chromosome ends are not eliciting reactions that would paste together the broken ends or checkpoint reactions that would stop the cell cycle. Telomeres also solve the end replication problem. So the inability of conventional DNA polymerases to deal with the very end of any linear molecule is compensated for by recruiting telomerase, this reverse transcriptase that carries its own RNA subunit that templates the addition of telomere repeats onto the end of the chromosome and replenishes DNA lost due to the end replication problem. So telomeres have been a very active topic of study in terms of cancer and aging, and I'm going to talk about some of the reasons for that. So in humans, telomerase is expressed in our germline and stem cells, but it's turned off in most of our somatic cells in early stages of embryonic development. So our telomeres are getting shorter and shorter with every round of replication, and that leads eventually to having a cell that has a telomere that's lost its ability to distinguish itself from a damage-induced DNA break end. And therefore, it elicits a cell cycle arrest. And this process is something that does occur during aging, and it occurs too early in some diseases where people have mutations in some of the telomere binding proteins or telomerase subunits. But those cells that have reached that point should be eliminated from proliferation. But if the cells have activated, have a mutation that inactivates the checkpoint response, they may go on dividing with this exposed chromosome end that's not a proper telomere. And for instance, the telomere ends can be fused together by a non-homologous end joining type process, for instance, and that creates a dicentric chromosome. And so when if the two centromeres are attached to microtubules going to opposite poles of the cell, that leads to stress along the DNA that's now stretched across the middle of the cell, and it's going to break at a random place. Sometimes it breaks many times. So DNA stretched across the middle of the cell that don't have the ends resolved, it is really problematic and could lead even to chromothripsis and real chaos in the genome. And that produces unequal segregation of the DNA. So you have cells that get an incomplete copy of the genome or extra copies of the genome. And usually this leads to genome instability and cell death. But it can also lead to new genomes that can be selected for under certain circumstances. And so for instance, in cancer cells, they may have gone through a lot of genome instability and have aberrant chromosomes, but if they find a way to re-stabilize the chromosome ends, usually they do this by reactivating telomerase. But if they don't reactivate telomerase, they either use alternative lengthening of telomeres, which is a kind of a recombinational way to use break-induced replication to copy whatever bits of telomere DNA are left onto all the chromosome ends. And as James said, one of the things that we found in our lab in Fission yeast, and we're looking at whether this occurs in human cells, is a way in which non-telomeric repeat sequences can acquire end protection capacity. So in fact, the ribosomal DNA can do that. And we're very interested in knowing if that happens in human cancer cells that are telomerase negative. But so telomeres clearly have a very central role in guarding the stability of the genome. And they both, losing telomere function can promote tumorigenesis by creating genome instability, but ultimately cancer cells that don't reactivate telomerase or have some sort of end stabilization will die off because the end replication problem and the genome destabilization will be too much. So here's just from the web a picture of a cell dividing where you see the spindle in green and the chromosomes in blue being pulled apart at mitosis. And here's just a diagram of what's happening. So the centromeres are, of course, being the first part of the chromosome that's pulled apart to the poles. And the telomeres are the last parts of the chromosome to segregate. So, you know, what can go wrong? I just told you that one of the things that can go wrong is that you can have fused telomeres that cause troubles at mitosis, can cause various catastrophes. One of the things that we've been working on in our lab is telomeres, the repetitiveness of them makes them difficult to replicate even by the conventional DNA replication machinery. And stalled forks often arise at telomeres. And if those don't get resolved, they lead to sort of entanglements that are not covalent fusions between telomeres, but entanglements involving single and double-stranded DNA. And exactly the structure of that entanglement determines how it'll segregate. And some of you may have heard Rishi Nagashan from my lab talk about this a few weeks ago in our departmental Friday research talks. But what I want to talk about today is a very different way in which telomeres can affect cell division at mitosis and meiosis. And that is one that affects the centromeres. So I'm going to tell you about a case, a scenario where telomere dysfunction leads to chromosomes that never even attach to the spindle because their centromeres have become dysfunctional. And so this story is going to start in Fission yeast, that's Schizosaccharomyces pombe, which is the organism that we've worked on the most in my lab over the last few decades. We love this model. The cells are easy to grow. You can manipulate them precisely, just like you can with Cerevisiae. You can do the airtight control experiments that are still difficult to do in human cells. There are only 3 chromosomes in pombe, which turns out to be quite an advantage for some types of telomere studies. And they have telomeres, centromeres, heterochromatin that are very conserved. The components and the principles of function are conserved with those of human, and actually more so than budding yeast. Chromosome dynamics and structure are conserved with humans. So it's a great model, but I'm going to end the talk by talking about human cells. So hold tight if you only like to hear about human cells. Okay, and I'm going to start by talking about meiosis. And just to, I don't think you probably need to hear this, but why should we be talking about that at a cancer center retreat? First of all, it's interesting. Meiosis, of course, drives sexual reproduction and promotes evolution. And it's sort of the raw materials on which we evolve. The study of meiosis has uncovered many principles that have illuminated mitotic cell cycles, so proliferative cell cycles. So things like the DNA repair pathways, much of what we know originated from studies of meiotic recombination and cohesion, principles of cohesion function, many of those started by studies in meiosis. And finally, and this is something that I'll come back to at the end of the talk, cancer cells often inappropriately express meiotic proteins and activate some meiotic processes. And so we really have to understand how those processes work in meiosis and how they work in mitosis when you don't have the full set of meiotic controls that apply to these processes. So we'll talk about that in a little while. So this is just a photograph of the former and current members of my lab that were at the last Meiosis Gordon conference. And so the work that I'm going to tell you about today, the story started with Michael Kletzstein's work when he was a postdoc in the lab. He has a lab now in Jerusalem. And Haitong Hao, who's a postdoc in the lab, who's here. And I will also be talking about the work of FD Kyriakou. So she's a postdoc that's now in Joachim Linger's group in Lausanne, Switzerland. So the story starts with looking at the function of this very dramatic nuclear reorganization that occurs when cells progress from mitosis into meiosis. So in mitotic interphase in Pombi, the chromosomes are arranged in the following way. The centromeres are all clustered in the vicinity of the centrosome that's sitting on the outside surface of the nuclear membrane that's shown in yellow here. And they're connected to each other by this conserved link complex. And the telomeres are in a couple of other positions. They cluster as well around the nuclear periphery away from the centrosome side. But when cells are induced to undergo meiosis, you have this switch of positions between the telomeres and the centromeres. So now the telomeres gather at this region beneath the centrosome and connect with the link complex, while the centromeres move away from this region. And I just want to plant the seed that there's a moment where the telomeres have started to gather at this region where the centromeres are before the centromeres move away. So there's actually about a half an hour of meiotic prophase where the telomeres and centromeres are communicating with each other. They're right next to each other. Okay, now this configuration where the telomeres are clustered near the centrosome at the nuclear envelope is something called the bouquet, and it's conserved throughout eukaryotes. So here's just what I think is a beautiful picture from Mays showing all the telomeres in green clustered to this limited region of the nuclear envelope with the rest of the chromosomes emanating therefrom, and that's why it's called like a bouquet of flowers. And in Pombi, we know how the bouquet is formed. It starts with the TAS1 telomere protein that we've worked on for so many years, and its interactor RAP1. So these telomere binding proteins interact with meiosis-specific proteins, bouquet 1 and 2, it doesn't matter the names, to connect them with the link complex. And because we know how the bouquet is formed, we can destroy it. And so we can delete any of the genes encoding any of these proteins, and you get the same kind of phenotype for what I'm going to tell you about today. And we call any of these deletions the bouquet delta phenotype. So what happens when we delete any of these genes and lose the bouquet? A lot of things happen, and we've published a number of stories about them. You lose the homolog alignment and pairing that you get that promotes meiotic recombination. You have problems with the partial nuclear envelope breakdown that allows for meiotic spindle formation. That requires the contact between telomeres across the membrane with the centrosome. But what I'm going to talk about today is this centromere story, a centromere maintenance story that we came at by looking at the function of the telomere bouquet. So I just want to start by showing you a movie of meiosis in a wild type cell when everything is going perfectly. So when you have, I'm just going to show you what the movie's going to show. So when you have bouquet formation, the centrosome for a couple of hours throughout meiotic prophase actually oscillates, migrates back and forth across the cell. And because all of the telomeres are connected to that moving centrosome, they're pulling the chromosomes back and forth across the cell, generating this elongated shape that you'll notice. And it's during that period when you have the real business end of meiosis. So homologous recombination is occurring during this time of movement. So that's the first thing you'll see in the film. Then the nucleus settles down in the center of the cell, it rounds up, and a spindle forms. Because these cells are small, the spindle will just look like one line, but actually it's many microtubules in the sort of basket-looking shape that you see in larger eukaryotes. And so you have then the two meiotic divisions, the reductional and equational divisions that you'll see that lead to an ascus containing the 4 haploid spores. So those are basically the gametes in Pombi. So here's the film. So tubulin is in green and chromatin is in red. So first you see this stage of prophase where the nucleus is being pulled back and forth across the cell via the centrosome, which you don't see here, and it's being pulled by cytoplasmic microtubules. Then it'll settle in the cell center. The microtubules go in the nucleus and make the spindles for meiosis 1, and do that again for meiosis 2. Okay, so you have this very orchestrated and neat meiotic program that generates the 4 spores, each of which have a perfect complement of the genome. So what happens if we destroy the bouquet? So a number of these things happen that I told you about, and I'm going to focus in on one of them in particular. And that's going to be illustrated in this film, where you're going to see that there's a centromere that you're going to have these spindles form at meiosis 1 and 2, and one of the chromosomes completely fails to attach to any of those spindles. The first thing you'll see is that the microtubules are going back and forth, and the centrosome would like to pull the nucleus back and forth, but there's no connection between the telomeres and the centrosome because we've destroyed the bouquet. And so the nucleus just sits there in the center with the membrane, which you can't see, kind of following the microtubules. And then you'll see meiosis 1 and 2. So here's the movement stage, but the nucleus doesn't move. The spindle forms here. Meiosis 1, one of the chromosomes does not attach, and it doesn't attach to any of the meiosis 2 spindles either. And so maybe I'll just play the film one more time because I just want to be sure you see it, that when the nucleus divides in 2, in fact, there's one chromatin mass staying in the center, and it doesn't attach to this spindle in meiosis 1 or 2. And this is something that we studied for a long time, these chromosomes that fail to attach. And what we found is the following. These chromosomes contain their centromeric DNA, but they have lost SEMP-A, the foundation of the centromere, and I'll talk about that more in a moment. They've lost all of the kinetochore proteins, and they've even lost the pericentric heterochromatin. So there's been a disturbance at the centromere that has propagated out for tens of kilobases, okay? So when these cells enter meiosis, something happens to the centromere. Okay, so just to talk a little bit about sort of a simplified view of what's at a centromere. So in Pombi, and this is something that's really conserved with mammalian cells as well, you have a central core of the centromere in regions surrounded by pericentric heterochromatin. So that heterochromatin is characterized by inaccessibility to transcription to a large extent, to histone H3 lysine 9 methylation, which provides a binding site for the HP1 protein. And this pericentric heterochromatin has a very important role in the establishment of what's called SEMP-A containing centromeres. So the SEMP-A nucleosomes, that SEMP-A is this histone H3 variant that's specific to the centromeres. And SEMP-A, in fact, forms the platform for the recruitment of all of the inner and outer kinetochore proteins that connect the centromere with the spindle microtubules. So something that's sort of was important for our studies is that it was known that pericentric heterochromatin, it's crucial for recruiting SEMP-A nucleosomes to de novo, like naive centromeres that have never been centromere sequences. But once SEMP-A chromatin is established, you don't need the pericentric heterochromatin anymore for maintenance of SEMP-A chromatin. So centromeres don't fall apart if you delete any of the genes involved in heterochromatin assembly or maintenance. However, what we found is that you do lose SEMP-A and centromeres if you delete heterochromatin genes in meiosis. And so that told us that there is a constant disassembly of centromeres in meiosis that makes them act like they are de novo naive centromeres. And this is all work that's published that I'm not going to talk about in detail. And basically what we found is that centromeres have a tendency to fall apart in meiosis. So this is a small proportion of the total centromeres. In Pombi, it's about 3 to 4% of all chromosomes lose their kinetochores when the cells enter meiosis if the bouquet isn't there. So the second part is that the telomeres, at this moment when the telomeres and centromeres contact each other, the telomeres create a microdomain that promotes kinetochore reassembly. And we actually know a lot about what the telomeres are doing to promote kinetochore reassembly. And that's not what I'm going to be talking about today as much as why centromeres would be falling apart when cells enter meiosis. This seems very dangerous. How and why would it happen? Okay, so to get at that, the first thing we did was look for mutations that rescue kinetochore loss in a bouquet deficient scenario. And so we looked at a number of meiosis-specific genes. This is a meiosis-specific phenomenon. And so in wild type cells, you rarely see what we call what we call kinetochore loss. We have an unattached chromosome with the centromere DNA, but without SEMP or the kinetochore markers. And so with all of the proteins that we've tagged in these cells, there's a very low level of this that occurs in a wild type background, whereas in a bouquet deficient background, it occurs in say 10 to 12% of the cells. And that's because there are only 3 chromosomes per cell. That means about 2 to 4% of all of the chromosomes. And that is suppressed specifically by deleting a couple of different genes. If we delete SPO11, that suppresses this kinetochore loss phenotype, or if we delete REC8. So what are those proteins? In fact, they're the foundational proteins of meiosis. Okay, so just to remind you about meiosis, so you start with haploid chromosomes, they undergo pre-meiotic DNA replication, and you have proper chromosomes in meiotic prophase that undergo meiotic recombination, and then at meiosis 1, rather than the sister chromatids being separated from each other, the homologous chromosomes are separated from each other, and it's only at meiosis 1, and only at meiosis 2 do you have an equational division that separates the sisters from each other, so you get 4 haploid gametes. Sorry. And what makes meiosis different from mitosis is this meiosis 1 reductional division where the homologs segregate from each other. And that requires a couple of things. It requires SPO11. This is a topoisomerase that generates the DNA double-strand breaks that initiate meiotic recombination. So they initiate, you have a break in one chromosome, it invades the homologous region on the other chromosome, and you get a crossover resulting in recombination. And crucially, you also need the meiotic cohesin, REC8, to allow this reductional division at meiosis 1. So cohesin are, of course, the rings that form around sister chromatids and keep them together just until the time that they should separate at mitosis, or in this case, meiosis. And a crucial feature of the meiotic cohesin, REC8, is that it does bind to centromeric regions, and it is retained through meiosis 1 to keep those homologous centromeres together and orient them towards opposite poles, but keep the sisters oriented together towards a single pole. And it doesn't get destroyed until meiosis 2. So SPO11 and REC8, the only proteins that we found were causing this centromere kinetochore loss in meiosis, those are the very foundational proteins of meiosis. So a couple of facts about that. For SPO11, we wondered, well, is it that SPO11 is creating a lot of double-strand breaks in the centromere region, and that's causing the centromere to lose its binding proteins? And that's actually not the case. So you can see here that deleting SPO11 suppresses this kinetochore loss phenotype, but replacing SPO11 with a point mutation that abolishes catalytic activity, it has no effect on the kinetochore loss phenotype. So catalytic activity is not involved at all. We also deleted a number of the genes that are required along with SPO11 to successfully make double-strand breaks, and none of them have any effect. Whereas a DNA binding domain point mutation does act like the null, the SPO11 delete, in suppressing this kinetochore loss phenotype. So you need the physical presence of SPO11 bound to the DNA, somehow causes sort of eviction of SEMP and the kinetochore, but its catalytic activity is not involved. For REC8, when you delete REC8, again, you suppress this kinetochore loss phenotype. There's another cohesin, REC11, that's involved with REC8 binding the rings that form around the chromosome arms, but not the centromeres. If we delete REC11, that does not suppress the phenotype. In fact, it makes it worse. Whereas if we use a TS mutation in PSC3 that abolishes REC8's localization to the centromeres but leaves the arms intact, you do recreate suppression of this phenotype. Okay, so it's specifically REC8 cohesin complexes bound at the centromere are causing centromere loss. Normally, at wild type levels of all these proteins, you need both SPO11 and REC8 to see kinetochore loss. But if we overexpress either of them, we see kinetochore loss in the absence of the other. And in fact, we don't even have to do this in a bouquet minus background. So I told you that colocalization with the telomeric microdomain reassembles centromeres if they've fallen apart. And that's why you normally don't have any problem with this centromere disassembly phenotype, because normally the bouquet is forming and the telomeres and centromeres will be in contact with each other and can reassemble the centromeres. But if you overexpress SPO11 or REC8, even in a wild type background, you see this kinetochore loss phenotype. So if you have too much of these proteins, the reassembly process cannot keep up with the disassembly process, okay? And I'm not gonna take the time to talk about it. We looked at a bunch of nucleosome remodeling complexes. So realizing that SPO11 and REC8 are destabilizing and in fact evicting SEMPE nucleosomes, we were looking at whether they do this in conjunction with various of the nucleosome remodeling complexes, and in fact they do. So they each require a specific chromatin remodeler, and I'm happy to talk to you, anybody, about which one of the remodelers those are and what that tells us about the process. So far I said SPO11 and REC8 are dangerous to centromeres, and meiosis is dangerous to centromeres. Both of them dismantle SEMPE chromatin utilizing chromatin remodelers, and the only reason that our centromeres aren't constantly falling apart during gamete formation is that the telomere bouquet is providing an environment to reassemble centromeres. Okay, now if SPO11 and REC8 can evict nucleosomes, we thought, well, if that's really true and they do it without all the rest of the meiotic proteins, expression of these in proliferating cells should cause kinetochore loss. And so we went ahead and looked at now mitotic cells, proliferating fission yeast. So in normal mitosis, now you're not going to see 4 meiotic products, but just the 2 mitotic products, 2 nuclei. And each of them will have a cluster of centromeres with their kinetochore proteins that we're going to be looking at. Sometimes there are a lot of things that happen in cells that lead to lagging chromosomes. So chromosomes that don't segregate right away, but they have their kinetochore markers. So just showing you now, this is in fixed cells. So normal mitosis, you see the spindle and the chromatin together in red, the chromatin in blue, a kinetochore marker, SEMP-C in green, and the merged image here. If you have a lagging chromosome, you see that one of these chromosomes has not segregated along with the others, and it has a kinetochore marker. It's not lagging because it's lost its kinetochore. So we're wondering if we can generate this new phenotype, a lagging chromosome without a kinetochore marker, analogous to what we saw in meiosis. And so this is just to show you that in mitotic cells, normally there is no expression of SPO11 or REC8. They are totally meiosis specific, but we can put them under overexpression promoters and see their expression in mitotic cells. And in fact, we do see this phenotype where we get chromosomes that are lagging, that are completely devoid of any kinetochore marker. And that is quantified here. So REC8 seems particularly potent at doing this in mitotic cells. And so because we could do this in mitotic cells, it allowed us to look into many of the details of doing a chromatin IP and seeing how much SEMPE is lost from every centromere and things like that. And I'm not going to talk about all of it because I want to get now to human cells. So SPO11 and REC8 have a tendency to dismantle centromeres. So they come along, SEMPE is lost in all the kinetochore proteins, and there's a disturbance that propagates along the chromosome for tens of kilobases and wipes out the pericentric heterochromatin as well. All right. So I told you meiotic genes are often expressed in cancer cells. So we wondered whether kinetochore loss could be an extra sort of burden of genome instability in those cancer cells that express meiotic proteins. And so we've started by doing some proof of principle type experiments to see if this might be possible. So now we're using U2OS cells, which I'll have more to say about in a moment. And we have integrated into their genomes an inducible promoter that controls either SPO11 or its catalytically dead or DNA binding dead alleles, as you'll see, or REC8. And in all cases, we have the protein tagged with GFP, so we can follow expression. And this is all under a doxycycline inducible promoter. And then, of course, we add dox, we induce expression of SPO11 or REC8, and then we, meaning Evtikia Kiriakou, did these experiments. And then she made whole cell extracts from these cells and did spindle metaphase spreads and looked at mitosis and how it goes in these cells. Okay, so first of all, just to show you that the inducible system works. So if we look at cells that are expressing the wild type SPO11, when we don't have doxycycline present, there's no band, but we see it. We have an anti-SPO11 antibody that we got from Dan Camerini at the NIH. And so we see that we do have, in fact, inducible expression of SPO11 for the catalytically dead and the DNA binding dead mutant as well. And we also have an anti-GFP antibody with which, again, we see that we have inducible expression. So normally, these proliferating U2OS cancer cells will not express SPO11 or REC8. And then we have REC8 here. Unfortunately, we didn't have an antibody against REC8 itself, but we had an antibody against GFP. Okay, so what happens when we express these proteins? So first of all, U2OS, as I hope that many of you will know, it's a commonly used cell line because you can make these integrations and grow the cells very easily, but it does have the defect that it's an alt cell line. Okay, so it is using this constant recombination of telomeres to maintain telomeres and to maintain viability and proliferation. So alt cells have some constant replication stress, and the telomeres are having a lot of stalled forks and a lot of entanglements. And so something that you see as a background in U2OS cells is a lot of chromatin bridges at mitosis, okay? And this is something that we also saw, and this is not affected by expressing SPO11 or REC8, okay? So these are just a couple of images showing what these chromosome bridges are looking like in our U2OS cells, but we don't see any significant increase when we express SPO11 or any of its alleles or REC8. So this makes sense. This is these chromosome bridges actually require that the centromeres are functional and are connecting to the spindles. That's why you get these stretched chromosomes, okay? And we don't expect this, it's the telomeres that are stretched and are defective, and that is not affected at all by SPO11 or REC8. In contrast, a phenotype that we expect on the basis of kinetochore loss is affected by looking at expressing SPO11 or REC8. So that is lagging chromosomes, like you see here. And we can't look at whether they have kinetochore markers in this system, or micronuclei. So both of these things, lagging chromosomes and micronuclei, these kind of defects are exactly what we would expect if we were seeing elevated rates of kinetochore loss when we express these meiotic proteins in U2OS cells. And here's the data showing you that the wild type SPO11 does increase the frequency of lagging chromosomes or micronuclei. The catalytically dead version of SPO11 does the same. And this mirrors what we see in POMBI, that the catalytic activity is not required for disruption of kinetochore function. The DNA binding to dead allele of SPO11 has no effect. It does not increase micronuclei or lagging chromosomes. And REC8, again, does affect it. So this is kind of amazingly mirroring what we see in POMBI cells. So the burning question then is, are these chromosome segregation defects that I showed you, the lagging chromosomes and micronuclei, are they caused by the kinetochore loss phenotype that we were sort of interested in looking at? And to look at that, what FT did was she made chromosome spreads and stained them for SEMP-B and SEMP-A. Okay, so what is SEMP-B? This is a protein that's present in mammalian cells that binds directly to the DNA at centromeres, okay? And it stays bound even in cases where SEMP-A and kinetochore function is lost, because SEMP-B binds directly to the DNA. So even if you have a neocentromere formation scenario, where there's a centromere formed at a new site, not the original centromere. SEMP-A is lost from the original centromere. It's only present at the functional centromere site, but SEMP-B stays at the original centromere. So what we chose as a kind of a proxy for kinetochore loss was to look for chromosomes that retain SEMP-B but lack SEMP-A. Okay, so, and one of the convenient things about this idea is that SEMP-B has this characteristic pattern of sort of staining on chromosome spreads, where it forms a bar across the two sister chromatids, and SEMP-A is in two foci, one on each sister chromatid. And you see that these kinds of bars of green staining, that's SEMP-B, surrounded by SEMP-A, are present at many of these chromosomes. And so to look for kinetochore loss then, we're scoring cases. And we're doing this all blindly. I'll show you how in a moment. So here's a case where you have a SEMP-B bar with a SEMP-A focus at either end. Here's a case where SEMP-A has been lost from one of the sister chromatids, okay? And then here's a case where it's been lost from both sister chromatids. And so what we did was find all of these SEMP-B bars without looking at any of the other channels. And Rahul Thadani in the lab wrote an algorithm for us to determine whether there were 1, 2, or 0 peaks of SEMP-A at each of the SEMP-B bars. And that data is shown here. So again, what you're going to see is that SEMP-B, the percent of kinetochores that have SEMP-B lacking SEMP-A is increased by SPO11 expression or the catalytically dead allele expression, but not the DNA binding dead allele. So that again is paralleling what we see in POMBI. And just to say that we expected the differences to be smaller when looking at single chromosomes than we do when looking at whole cells, as in the lagging chromosome and micronuclei assays. And we can talk about why later if anybody wants to. And so REC8 also has this effect. So SPO11 DNA binding does lead to kinetochore loss in these cells, as does REC8, these meiosis-specific proteins. Okay, so to bring together what I've told you, centromere maintenance is really precarious if SPO11 and REC8 are present. So they come along and they evict SEMP-A nucleosomes and surrounding heterochromatin, but in wild-type POMBI cells during meiotic prophase, the centromeres and telomeres are together, and the telomeres provide this microdomain that promotes centromere reassembly. And we know that what telomeres do is they provide their own heterochromatin in trans that substitutes for the pericentric heterochromatin. And that's allowed us to determine what heterochromatin was doing anyway to promote SEMP-A centromere formation. It involves kinase cascades involving the aurora kinase. And we can talk about that another time. And so the telomeres help to reassemble centromeres. And I didn't draw reassembly here, but you can imagine SEMP-A reappearing here and the heterochromatin being reassembled. And I sort of didn't draw it because there are many questions that we don't know the answers to, whether the presence of the telomeres there, that you're having constant eviction and reassembly, or whether they're just stabilizing the region so much that you never really lose the SEMP-A, but that's happening. In cancer cells, there's no telomere bouquet. There's no contact between telomeres and centromeres. There will be no rescue of any centromere loss, kinetochore loss that occurs. Okay, so just to finish, meiotic centromeres can fall apart, and even mitotic centromeres are not rock stable. And I should have said that it's been considered in the field that SEMP-A nucleosomes are rock stable, and that they are inherited through many generations. And that they basically don't turn over. Well, in fact, they do turn over if SPO11 and REC8 are present. And one of the things that we're starting to look at are the mechanistic details of how that works. SPO11 and REC8 destabilize SEMP-A nucleosomes as well as the pericentric heterochromatin. So one aspect of this story is that it could explain that there's yet another danger to meiosis and fertility. So something that's been seen in both mouse and human oocytes from females of advanced age is that there are hallmarks of kinetochore loss occurring. And so I think that our observations could provide an explanation for that, that you have these meiotic proteins present, and they do provide a danger to centromeres and kinetochores. And finally, we showed that SPO11 and REC8 compromise human centromeres, not only POMB centromeres. And since there are a number of cancers like melanomas that express these proteins, we wonder if this could be a point of vulnerability for those cancers, that kinetochore loss is something that's going on, and that's something that we're starting to look at. So I think I've told you about the people doing the work, and I thank you for listening to me. So I should exit.
Great. Thank you.
I went over time. I didn't even know.
We have time for maybe 1 or 2 quick questions. And just to remind people, if you have a question, just put your name in the chat box and I'll call you in the order that your name appears. And so far, let's see, we have James have a question. Go ahead, James.
So first off, great talk. That's really exciting. And so it's what I found interesting is that you have 2 to 3% kinetochore loss even in the wild type. It would sort of suggest that either evolution hasn't been perfect, or there would be a further cost to making that better, or there's actually some benefit to the phenotype.
Yeah, so that's a great point to make. First of all, just to ask, are you talking about the POMB case or the human cell case?
Well, the POMB, but I imagine it applies probably for the human cells as well. But
well, so it is. So first of all, I should say that when you tag multiple proteins in a cell, so if we tag a kinetochore protein and also tubulin, so we can view the spindle, and also the centrosome proteins, we do see. Usually when we tag just one protein, the cells are totally fine. And if you do any sort of gross analysis, everything is fine. But we see this kinetochore loss at maybe, as you're saying, a couple percent when we have all these proteins tagged. And that could be part of the reason that we see a bit of kinetochore loss. But I think your point is really important. And I sort of meant to put it in the discussion point, the perspectives that there is a lot of remodeling that's occurring at the centromere during meiosis. So you have to change over from having equational segregation to reductional. And you have to replace the cohesins, the mitotic cohesins with meiotic cohesins. And there are all these things that have to occur. And so maybe it's built into the system to have sort of a bit of kinetochore loss, and it's safe because you know that the telomere bouquet is going to be there. So the cell can tolerate kinetochore loss. It's actually, it's just not a problem because in wild type cells, there will always be a telomere bouquet.
It's good enough.
But yeah, that the destabilization of the kinetochore, in fact, the outer kinetochore is something that does disassemble during meiotic prophase anyway. And I guess what was new here is that the actual whole inner kinetochore and SEMP-A is disassembling. It seems dangerous, but I guess it's not so dangerous because the telomere is always there. Sorry.
Yeah, thanks.
So the next person's Allison. In the interest of time, do you have a really quick question, Allison?
It'll be pretty quick.
Yeah.
So yeah, my question's on the SPO11 catalytically dead mutant. Like, what do you think SPO11 is doing if it's not like the double-strand break activities?
Yeah, no, that's a great question. So yeah, and it's something that I really didn't mention and is very important. So SPO11's real function in cells is to create the programmed double-strand breaks that are required for meiotic recombination. And this is a very controlled system during meiosis, but of course could be very dangerous if expressed in mitosis where you don't have all the other checks and balances. And perhaps, so we know you don't need the catalytic activity to evict nucleosomes, but nucleosome hotspots for DNA double-strand breaks are in fact at nucleosome depleted regions. And we know that SPO11 requires chromatin remodelers in order to disrupt the kinetochore. And so it may be that sort of nucleosome eviction is part of SPO11's function, but that SEMP-A nucleosomes are just particularly susceptible to this nucleosome eviction. And you know, it's maybe the tip of the iceberg in terms of the genome becoming generally sort of loosened when you have these kinds of proteins around, and how many proteins act in that way. But it could be that just the DNA binding by SPO11, and we don't know if there's breathing always on the nucleosome, and if SPO11 happens to get in there, you can't sort of reassociate the nucleosome, or if it actually is bringing a chromatin remodeler that's clearing out the nucleosomes to allow SPO11 binding. So those are all possibilities.
Thanks, Julie. It's really interesting. I'll look forward to talking to you more about this stuff.
Great. Thanks so much, Julie. And I'm going to hand it over to Craig to introduce the next speaker.
Thank you.
Thank you.
Good morning, everyone. So by way of introduction, I'd like to just give you a little bit of context. So if you are anything like me, you assume that basic cellular functions are essentially very well conserved amongst all of us, irrespective of our genetic heritage or specific nuances related to race or ethnicity. Things like telomere function, for example, and similar cellular functions, I think, certainly I have always assumed were so well conserved that you really didn't need to think about how all of us may differ a bit with respect to these pathways. And I think it's becoming quite clear that that's a flawed premise. With respect to at least some cellular processes, there clearly are very important differences that can impact our research in a variety of ways, whether it's through direct mechanisms or more subtle genetic modifiers. It's very clear now that genetic ancestry and background can have a major impact. And so I think it's actually beholden on all of us as we think about this, whether you're working in human cells directly or in model systems, to take these considerations very seriously as you think about the questions that you're asking. And I would point out that this is actually something that's been formally acknowledged in recent years by the NCI, and there are specific grant mechanisms that have been put forth, some very recently, in fact, to encourage everyone, even at the most basic level, to think about these kinds of issues. And so it's very timely and important to bring this to bear. And in organizing this retreat, we felt it was important to have this discussion. And very fortunately, we have the perfect person to introduce the topic for us. Our next speaker is Dr. Evelyn Barreo. Evelyn's been a faculty member at both the University of Colorado as well as Colorado State University for over 20 years, where she's risen through the ranks to be a full professor in both the Department of Psychology as well as the Colorado School of Public Health. So she has extensive experience in considering the disparities and the differences that exist amongst humans of different backgrounds. And we're very fortunate to recruit her just this past year to lead this effort in the University of Colorado Cancer Center as the director of the Office for Community Outreach and Engagement. So Evelyn, thank you so much for being here today, and look forward to hearing your thoughts.
Well, thank you very much for having me. And as you all realize, my background is significantly different from yours. So I don't pretend to know your science, but I do know mine. And so my goal is to kind of show you how cancer is affecting Coloradans. And let me share my screen before I get too ahead of myself. Okay, can everybody see that? You can just give me a thumbs up if.
Yeah, looks great.
Okay, great, thank you. Yeah, so my presentation today is going to be talking about the cancer burden, risk, and disparities in Colorado, perhaps presenting some population health data that you might have not seen. And then my intent is that this would lead to discussions from here on as you talk about your research, not only as a group, but also in your labs as you start thinking about your research. As Dr. Jordan mentioned, I lead the Office of Community Outreach and Engagement, and the office was established just last year as a result of the NCI's interest in addressing disparities more comprehensively. So there are three aims that the office has. One is to provide you with comprehensive up-to-date data on the cancer burden, risk, and disparities that affects what we call our catchment area. And for our cancer center, the catchment area is the entire state of Colorado. Other cancer centers just have a section. For example, California may just do Southern California or the Bay Area. For us, it's the state of Colorado. Another goal of the Office of the COE is to help enhance and disseminate the research that you do for impact so that we can reduce the cancer burden, risk, and disparities, and to engage our communities in science, education, and advocacy. So those are our aims, and the reason I am here is to present perhaps number one, which is, so when we talk about the cancer burden, risk, and disparities, what specifically are we talking about? And hopefully lead you with a desire to enhance your research to address those disparities. So the NCI defines cancer health disparities as the differences in incidence, prevalence, mortality, and burden of cancer. But it's not just differences, it's differences when they are related to the adverse health conditions that exist among specific groups in the US. Basically, those adverse health conditions is what affects what we call disparity populations, the racial ethnic minority status of individuals living in rural and frontier counties, those who are poverty-stricken, children, older adults, and LGBTQ+ are just examples of individuals that suffer from disparities. And when we talk about disparities and those differences, we are not just talking about numbers. So for example, if you look at this graph, it kind of shows you what is the cancer incidence and mortality from the various cancers. And from this graph, you might assume, well, you know, whites overall have higher incidence and mortality from cancers in some cancers than other groups. But if you actually start looking at the subgroups, then you really start looking that relatively to other groups, to the white population, this when you start seeing the disparities. And my hope is to show you how in Colorado, we have looked at the various segments, the various disparity populations to assess what is different and why is that, or why it may be different. So this other graph kind of shows you that overall, the top incidence in the US and Colorado are similar for the various cancers, the top 10 cancers. And you may not be able to see here, but there is an asterisk for non-Hispanic white populations. For Colorado is actually lower for most cancers. Colorado is the black. Oops, I'm sorry. Colorado is represented by black, and the US is represented by the gold. And if you can see for non-Hispanics, whites, the incidence is actually lower. But when you look at the Hispanic whites in Colorado, you actually see, and there is that asterisk that is reversed, that compared to Hispanics in the US, there are this, significant disparities for breast, lung, and colorectal cancer in terms of incidence. And you see that same pattern for mortality, where, you know, the US fares worse than non-Hispanic whites, but when you look at Colorado Hispanics, they have higher mortality from lung, breast, and colorectal cancer. So it is when you actually start looking within populations and compare them to a relatively comparable group is when you start seeing those disparities. So disparities is not always just in relationship to the non-Hispanic white. It could be within groups that you see differences. And in Colorado, we see that for Hispanics. So I'm going to be using the Hispanic population and some of the disparities that affect them as an example of the cancer disparities in our catchment in Colorado. So just to summarize, we see here that there are ethnic disparities in Colorado by incidence and mortality of the top 5 cancers, but particularly significant for lung, breast, and colorectal cancer. If you actually start looking at where those individuals are, because of population density, we know that the majority of Hispanics live in urban areas, and this graph basically demonstrates that there is a higher percentage of late stage diagnosis among Hispanics who live in those urban areas. And that includes the Denver metro area, Greeley, Colorado Springs, and Pueblo primarily. You see that same pattern for colorectal cancer, where you see primarily the urban areas where you would see the late stage disease for colorectal cancer for Hispanics. There are also differences based on location. And in Colorado, rural and frontier counties have the highest number or higher incidence, mortality, and late stage disease than the more urban counties. And what you see with this graph is that the darker blue represents worse disparities, and this is provided by the Colorado Department of Public Health and Environment, in which they have combined incidence, mortality, and late stage disease for the three cancers that I mentioned: lung, breast, lung, breast, prostate, and colorectal cancer. But if you actually look closer to what may be explaining those disparities, if you look at this green map with darker meaning higher population density, and the lighter means lower population density, it is those frontier counties that you see in the southern Colorado where you see the worst disparities. The teal dots represent where at least 25% of the population are Hispanic. So Hispanics are not only concentrated in the metro areas, but they're also in rural areas of Colorado, and that's where we see worse disparities. So if you were to kind of overlap those two maps, you will see a correlation of worse disparities with rurality, with poverty. The lines indicate poverty, where at least 15% of the population are under federal poverty levels, and you see the teal dots being Hispanic. So that's why we say that cancer disparities are more pronounced for Hispanics who live in poor and isolated counties in Colorado. So what I would like to provide is an overview of what we know, and we know quite a bit about lifestyle risk factors for cancer. And this data kind of shows you the role of diet in common cancers. This comes from the American Cancer Society. I don't see the reference there, but that's where I got this information, where we know that it increases alcohol, excessive caloric intake, and weight gain increases the risk for breast cancer. For colon is red meat, same from prostate, and alcohol and sodium for GI cancers. And we have some evidence that suggests that higher intake of vegetables, reduced alcohol, decreases the chances of some of these main cancers. We also know that obesity plays a role, is a risk factor for women for endometrial, ovarian, colon, and breast cancer, and renal cancer, for men for colon and prostate. And we have some understanding of what may be the mechanisms behind it. Hyperinsulinemia is especially important because it concentrates, is a concentration of central adiposity that is associated with cell growth and its proliferation. I would imagine some of you are doing that type of research. And we know that adipose tissue is primarily the source of estrogen, which has been linked to the carcinogens that lead to some of these cancers. We also have some evidence that physical activity, although it's not as strong as for the other risk factors, we are not sure if some studies show an association and others do not show that association. We know for colon cancer that 30 to 40% of, there's a 30 to 40% decrease in risk among men and women who engage in physical activity. And for prostate cancer, the findings are inconclusive. And there are some possible mechanisms as to why physical activity may play a role. We know that among those who are active, it decreases the GI transit time, which decreases the carcinogen exposure. It enhances immune function with moderate physical activity, and we also know that it lowers the levels of reproductive hormones. And we are still trying to understand the role of physical activity, but overall, this is what the evidence suggests. We also know that disparities are due to low cancer screening rates and the lack of timely treatment among some of the disparity populations. And this is just to show you at the national level among Hispanics and cancer screening. We know that among men and women who had a colorectal cancer screening, you see that whites, represented by the red graph, have higher percentage of cancer screenings compared to Hispanics that are the purple line. Same thing, if you actually start looking by income, you see that actually is not all Hispanics, is mostly Hispanics of lower income who are driving those statistics compared to Hispanics of higher income. They actually mirror more the rates for non-Hispanic whites when you consider the role of income. Same thing with cancer treatment. Of those who have been treated for breast cancer within a year of diagnosis, you see the darker blue, a higher percentage of whites, a much lower percentage of blacks. Of blacks and Hispanics. When you actually start looking within the groups, there's a lot of variability. There in Colorado, we have primarily individuals of Hispanic descent that are of Mexican background. Then there are also disparities in access and quality of care. We know some access barriers such as insurance is behind some of the disparities that I just covered, where we know that there's a higher percentage of uninsured Hispanics, which is the green, and then a lower percentage of Black and whites who are uninsured in the US. Also, having a usual source of care due to financial or lack of insurance, we know that there's a higher percentage of individuals who are Hispanic who don't have a usual source of care compared to the other ethnic groups. When you actually start looking at the role of insurance, then you see that it is Hispanics who don't have the insurance compared to those who do. Access barriers, those Hispanics, adults 18 to 64 years old who needed care, but sometimes or never got care. You also see that, and those are significant. There's a higher percentage of Hispanic compared to white, for example. And you also see the same role of insurance among Hispanics, with those who need care but never got it is higher among uninsured Hispanics versus those who have private insurance, for example. And same thing with quality of care. I will actually just move quickly through this because we're short of time. Let me talk about the cancer burden risk and disparity specific to Colorado. So this is a summary of the data for Colorado, and this is doing population-based research looking at the cancer registry in Colorado. And we know that, as I mentioned, there are worse disparities in rural areas by incidence, mortality, and burden. As I mentioned, for Colorado Hispanics, lung, breast, and colorectal cancer. And when there's poverty, there's an intersection between poverty, rurality, and Hispanic ethnicity. Where they intersect is where worse disparities exist. And then these are the top mortality and incident cancers in Colorado. Melanoma is what is different from the rest of the country, in which is the 5th incidence, but the incidence of mortality is higher in Colorado than the US. And here are some of the lifestyle risk factors that I mentioned. HPV is one that I did not cover, but HPV is quite low among young adults and in rural areas. And there are some statistics there on the low breast cancer screening rates by poverty, by Hispanics, by rurality. Same thing with colorectal cancer screenings are much lower for those disparity populations in Colorado. These are called our priorities. These are the Cancer Center, the University of Colorado Cancer Center priorities, because of what we see are disparities in Colorado compared to either non-Hispanic whites or Hispanics across the country. So why is this all relevant to you and to perhaps all of us? And this is a position statement from the American Association of Cancer Research, the American Cancer Society, the American Society of Clinical Oncology, and the National Cancer Institute, where it says that given the looming crisis in cancer incidence and mortality disparities that affect minorities and the medically underserved, it is our collective hope that in this period of cancer research, when significant breakthroughs are being discovered, that there will be opportunities to apply this new knowledge to all populations and thus eliminate cancer health disparities for current and future generations. And if you remember our ads, we are branding ourselves as conducting breakthrough research. And this is exactly what this is referring to, that while we conduct breakthrough research, we need to do it in a way that affects all populations. So some of the goals for cancer disparities research, and when I talk about cancer disparities research, some people think that somebody needs to have a particular expertise in cancer disparities, and that is not the case. We can all do cancer disparities in the research that we do, in how we apply our research, without necessarily having an expertise on cancer disparities. For us to address the disparities in our catchment in Colorado at the Cancer Center, we need to be conducting groundbreaking mechanistic research that leads to groundbreaking scientific discoveries to reduce cancer disparities. That is talking about you who are doing molecular and cellular at the very mechanistic level. We need to be conducting that kind of research. One of the reasons why I didn't present the research is because we don't have as much. We have some papers published, but we don't necessarily have a cohesive disparities science that could be presented. I'm not saying it doesn't exist, but we have more research to understand the population level disparities and the risk factors. And then those who do translational research needs to translate those discoveries into approaches and products that can change the paradigms of cancer care to reduce the disparities in our catchment in Colorado. And then some of those who are population scientists need to be conducting groundbreaking research that prevents cancer, promotes equity, and shapes the policy and the healthcare to improve the cancer outcomes that I just went through. So these are cancer disparities that affect Coloradans, and we need to be addressing those disparities. Recently, there was a report on cancer disparities at the national level that compared 30 years ago to now, and the disparities unfortunately continue to be the same or continue to be worse. So if we don't start conducting this kind of disparities research, another 30 years might pass and we may be just kind of presenting the same data or worse data. So that is basically the end of my presentation, and I'm happy to contemplate questions. And I don't know what time it is, so I'm gonna.
Yeah.
10:15.
Thank you very much, Evelyn. You know, it's worth giving a specific example because this is certainly something that got my attention, something I've learned from interacting with you in the past year, Evelyn. And that is that despite, as you nicely articulated, the access to care is a huge factor that can affect disparities. But even, and this is pretty recent research, even when you normalize for that, if you take into account relative access and so forth, there can be major differences between the way, for example, Black and white patients respond to chemotherapy. This is in my own work in leukemia. There's been studies that are actually just coming out this year that says that in association with specific mutations, and again, normalizing for other socioeconomic factors, there are major differences in the way that patients respond as a function of genetic factors that can be traced back to ethnic background. And so I think especially for this group, that's where I certainly was really quite shocked to see that, and it really sort of brought it home. I'd also like to just mention for this group that as a consequence of our interactions with Evelyn, it's motivated us to try to bring resources forward for the laboratory community to focus on this. So the Colorado Center for Personalized Medicine has begun an initiative where they capture blood sample from pretty much every patient who walks through the door of the University of Colorado Hospital. So it's a very large scale effort to do broad GWAS-based studies. And so they have a high density STIP array, and they're actually in the process of capturing a very large number of patients, which will of course include a number of people, different ethnic backgrounds. And so this is a resource that we hope to be bringing online in the next year or so, where you'll be able to mine that data with respect to any particular pathway of interest to determine whether or not people with specific ethnic backgrounds have differences that may be scientifically important. So with that, I think we could just take a couple of minutes for questions. Anyone's comments? I'm just reading from the chat box. So here's a question from Jamie Stutz. Evelyn, recognizing that there's almost always a need for more research, do you see particular opportunities or research gaps that we need to fill to address some of the disparities that you've outlined? Perhaps areas of disparities where we would not have an answer for a question like, what are you doing about X or Y disparity?
Particular opportunities or research gaps. I think we have both. We have research opportunities and gaps, in the sense that we really don't understand, at least at the mechanistic level, that is what the MCO investigators, there's still a lot of questions. Maybe there are no differences at that level. So, but we don't know that. We don't have good and complete science until we have answers to those questions. So there's an opportunity there, but also a gap in terms of understanding what drives disparities. If we were to have those answers, then that could lead to the translational research, how we do translational research. As Craig mentioned, are there differences based on the biology of individuals, based on their race, ethnicity, that explains differences on how people respond to medication or treatment, cancer treatments? Hopefully that answers your question.
Yeah, and James, you had a quick one here?
Yeah, just quick. I mean, I guess one concern, by a great talk, Evelyn, but would be with, with research that's developing new therapies like, immunotherapies and targeted therapies that are way more expensive. And is that only going to increase disparities, particularly based on income?
Yes, and that's why this particular slide here at the end just kind of really talks about how it is a puzzle that we all need to be part of, because that is exactly what would happen with increased cost of treatment. It increases those disparities. So those individuals that look at that, Kathy Bradley being one of them, who looks at the financial implications of treatment for patients, we need to be shedding light on how that contributes to disparities. But honestly, the gaps are more on conducting that groundbreaking mechanistic research. That's where most of the gaps quite honestly are on cancer disparities research. So you have endless opportunities to address that. And the NCI, I've seen an increase in RFAs on cancer disparities.
Thanks.
So Debashis Ghosh, who's our colleague in the School of Public Health, asks, Evelyn, you've shown a lot of population level data in terms of cancer incidence. What's available in terms of studies that enroll and involve Colorado-based participants?
Are you referring to cancer patients when you talk about cancer participants, or a patient? Are you referring to patients?
Yeah, I mean, either way. I mean, it could be in the community or could be in the clinic. Yeah, either way.
Yeah, that is actually, I should have just assumed that the answer applies to both. We do have an underrepresentation of individuals of ethnic minority backgrounds, of rural populations, of LGBTQ. Some of the disparity populations that I listed at the beginning tend to be underrepresented in the research that we conduct. And there are various reasons, but some of the reasons is we don't do enough community outreach and engagement, and that is the charge of my office, is to help the Cancer Center to better engage and conduct research in those populations.
So maybe just one more in the interest of time here. Jack Nikoloff asked, in your early slide showing disparities among ethnic groups, are these data age adjusted? Can some of the difference be explained by different lifespans since cancer hits elderly more frequently?
Yeah, potentially, but when we have control for some of those variables, we continue to see those disparities.
Great. And again, in the interest of time, I think we're going to need to move on. Thank you again, Evelyn, for a wonderful talk.
Absolutely. Thank you for having me. Let me take out my
So
sharing screen.
perhaps we can break for 5 minutes and then come back at 10:26. And it's my hope that, take bio breaks and so on, but you stay on and keep talking to each other, even during break, because this is sort of what we'll do if we were meeting in person for the retreat. And also want to alert Julie, there's a couple of remaining questions in the chat. If you want to go back and address them during break, it's up to you. So thank you. We'll see you back in like 5 minutes.
Thank you.
Hi, Julie.
Hi, Isabel. Yeah, I just answered one of the questions in the chat box, but no, that was to Debashis. I don't know if
this is Isabel Schlapfer.
Yeah.
And I enjoy your talk. I don't think about the kinetochore and and the telomeres, and much less about the bouquet. That was super cool. Thank you. But I study cancer cells, and I do see some of these MPA, B, and others come up in my RNA-seq, and I have to ignore them a little bit. I don't ignore them. I just I'm intrigued, but yet very confused because, like you said, is meiosis, and these are cells that went rogue.
Are you seeing expression of meiotic genes in the cancer cells? Is that what?
Yeah, some of them. Oh, I got my video on. Yes, I do. Yes, some of them. And now your talk made me think I should go back and look at them, because why are they there? These were terminally differentiated epithelial cells that went, that reverse a little bit, but not completely, not to the point of having meiotic proteins. So your talk was very intriguing. And the fact that you mentioned these two, ESPO11 and the REC8, as evicting the SEMPA out of the central, they don't allow, they evict the SEMPA out, so the centrosome integrity.
Yes, the kinetochore integrity is lost.
Exactly. So how do they get regulated? My question, they seem so critical. What makes them come on and do their job? What's the signal?
That's a great question. It's just an observation that they are expressed in some of these cancer cells. And so I don't know if it's that, normally the cell has a very tight program to keep meiosis genes repressed when meiosis isn't occurring. And so, is it that system has been, that the repression, that the heterochromatin that represses meiotic specific gene expression is screwed up? Has there been selection in the cancer cell line for expression of these genes? There are cases like, it's been shown in some of these alt cell lines that use telomere recombination to maintain telomeres, that they express synaptonemal complex proteins. So proteins that are involved that are meiosis specific, that are involved in the homology search, because these telomeres have to find each other to undergo recombination. And so actually expressing meiotic genes is an important part of their survival strategy. So I don't know if there's, so one of the things we'd like to do is we want to look in more cell lines, and we sort of did this with only a single level of induction of SPO11 and REC8. So there's a lot of things to do, but to find cancer cell lines that we can grow that do express these genes, see if they show signs of kinetochore loss, see what happens if we knock down those meiotic genes or knock out the meiotic genes that shouldn't be expressed anyway, and see what happens to those cancer cells.
Yeah, no, that's what I'm going to do is look at my gene list and go to those, you know, the genes that I didn't understand that are important, they're there and significant, but what do they do? So I appreciate your talk very much, and perhaps I could email you with the goodies I find.
Yeah. So where are you? Are you at Anschutz or in Boulder?
Yes,
or?
I'm in Anschutz. I'm in medical oncology, but I'm a basic scientist. So I do hormonal cancers, but I focus on metabolism, lipid metabolism. So yeah, that's why when I have to make the connection with the kinetochore and the centrosomes, that makes my head spin. But it doesn't mean it's not important. It means that I understand.
No, that's great. And we also have some reasons to think about lipid metabolism, which I never thought about. So maybe there's a lot to talk about. Are you you're just one floor above or below?
Yes. And I see Dr. Sclafani, who is a yeast expert and hello, I haven't seen him for a long time, but I trained with, I don't know if you met Dr. James Mattoon. He's deceased now. He actually died of prostate cancer, but he was at CU in Colorado Springs. He was the big yeast geneticist, and I know Dr. Sclafani knows him. So he was my mentor a long time ago. So I started doing Saccharomyces cerevisiae work with the heme and the aminolevulinic acid and all that porphyrin. So I was I do know how yeast cells. I mean, it's been a long time, but there's such a great model to study this. So anyway, just wanted to say that. Thank you so much.
That's great. Thank you. Nice to meet you.
Okay, hope everyone's back and ready for the second half. So I want to start by telling you that we had excellent submissions for talks from the trainees. And we were able to, even though we only had 2 talk slots, we picked 5 of them for Trainee Career Development Awards of $2,000 each. And I want to congratulate the 3 awardees who will not be talking. They are Afandi Triciani, postdoc at Anschutz, Bianca Nold Lopez, an undergraduate researcher at Anschutz, and Edma Ivanchovi, a postdoctoral fellow at UC Boulder. So let's have a round of applause for those excellent researchers, trainees. All right. And we do have 2 talks from the trainees, and I'm pleased to introduce the first one. Lydia Hisley, a postdoctoral fellow at Colorado State University, is going to talk about punctuated aneuploidization of the genome. So go for it, Lydia.
Okay. You guys can hear me, see me? Good. So this is a little bit of a different title than what I originally submitted, because I decided that I would combine 2 stories that have recently come out from the Argueso lab. So I'm a postdoc in Lucas Argueso's lab up in Fort Collins. And in the Argueso lab, we fully appreciate that genomic mutations are essential for the evolution of biological systems. And we don't focus on all mutations, although we think about them quite a lot. We tend to think about structural alterations the most. And examples of structural alterations that are pertinent to probably everybody in the audience are loss of heterozygosity, which can arise from mitotic recombination to heal double-stranded breaks, copy number alterations that result in duplications and deletions of genomic loci, and then whole chromosome alterations, which are classically known as aneuploidies. when I joined the Arguesa lab a couple years ago, I came in really wanting to understand a couple key aspects of genome biology and genome evolution. And they fall into the questions of how do structural alterations arise in the genome? How do they contribute to the overall structure and function of the genome? And how does structural genome evolution modulate the phenotypic and fitness potentials of the organisms that bear these genomes? And so today I'm going to tell you the work that we've done to address this first question, how structural alterations arise in the genome. And I'm going to focus mainly on the two types of structural alterations of whole chromosome gains and losses, and loss of heterozygosity resulting from mitotic recombination. So Darwinian principles tell us that mutations occur in the genome slowly and gradually over long periods of time. And this gradual process and gradual ideal has really framed how we interpret genome evolution over a very long time. And we know that it happens, but gradualism actually fails to explain some accelerated modes of evolution that we've started to see. And this is probably best embodied in the development of cancers. So by the time they're detected, cancer genomes are riddled with complex patterns of karyotypic alterations. This can range from aneuploidies, like extra copies of chromosomes, missing chromosomes, to large gross chromosomal rearrangements and duplications and deletions. And this connection between the disease state of cancer and chromosome abnormalities has been well observed for over 100 years now, but actually the drivers that underlie this evolutionary process remain fairly unclear. And moreover, we know that the rates that structural alterations like aneuploidy and loss of heterozygosity occur are quite rare. And so it remains unclear how gradualism alone could drive a process this remarkable. And so one of the questions I've been working on in the lab with the rest of the lab members is trying to understand how do cells actually develop these very intricate karyotypes. People have put forth numerous models over many years now to explain this accelerated evolution that we see in cancers. Probably the most well-known model is called the mutator model. And this posits that after acquiring a destabilizing mutation, cells can enter kind of a chronic phase of genomic instability and acquire new mutations at accelerated rates. With regards to whole chromosome gains and losses and loss of heterozygosity, you're probably most familiar with the term chromosomal instability, or SIN, which describes a mutator-like state of chronic genomic instability that results in increased rates of whole chromosome missegregation and increased rates of loss of heterozygosity. Another model that is emerging and that we have started to look at ourselves is this model of punctuated bursts of mutation. And this derives from Stephen Jay Gould's and Niall Eldridge's theory of punctuated equilibrium, which basically says that cells can exist in a state of genome stability for very long periods of time, and then in a short burst of instability acquire multiple mutations simultaneously, after which they resolve this instability and they go back to a phase of stasis. And this, actually, when I joined the Arguesa lab, I'd never even heard of this mode of evolution before. But in the last 10 years, numerous studies using genomic approaches to analyze tumor subpopulations and tumor genomes have started to point to this mode heavily for the acquisition of early mutations and early chromosome alterations in the development of cancers. So again, the question we're going to talk about for the rest of the talk is, with what dynamics do structural alterations arise in the genome? And can we differentiate between these three models to understand how cells get structural alterations? And we also use yeast. We use the budding yeast Saccharomyces cerevisiae to look at this for a number of reasons, which I just briefly want to go over. Dr. Cooper did a very nice job already explaining this. We use the S. cerevisiae genome, the budding yeast genome, because it is among the best understood and characterized eukaryotic genomes. It's 250 times smaller than the human genome, which makes it much easier to work with. But despite its small size, it is still quite complex. It has discrete chromosome pairs, and these chromosomes have a lot of the same structural complexity that human chromosomes do, including centromeres and telomeres and repetitive elements. They have transposons that can mediate genome evolution over time and copy number variations that are intrinsic to the genome. And we also like to use Cerevisiae because these pathways that maintain genome stability in yeast are highly conserved across eukarya, including human cells. And so using yeast, we can engineer yeast so that we can use selection-based assays to recover clones that harbor specific mutations. And we can do this even in extremely small populations of cells. And then we can take these mutant clones and we can sequence them, whole genome sequencing, for very little cost at very high coverage. So this allows us to really comprehensively characterize how structural variations accumulate in the genome, which would be very hard to do in a bigger genome, like a human's. And so using yeast, we tested the question of whether gradualism can account for the acquisition of structural variations in the genome. And we, operating under Darwinian principles, we could literally just follow a prediction that the rate at which two independent mutations should arise in a genome should be the multiplicative product of the rate at which each individual mutation is acquired. And so we designed diploid yeast strains such that we inserted counter-selectable markers at two individual loci, which is indicated in this figure by these stars. And so each of these markers can be selected against using specific drugs. And so essentially we could take a population of these cells and we could plate it on to media containing one drug that would select for the loss of one marker, on a different drug to select for the loss of another marker, and on media containing both drugs, so that we could recover clones that had lost both markers at the same time. And we use fluctuation analysis to do this. And what I'm showing you right here is a schematic for the loss of heterozygosity assays that we did. And what we found was that we recovered double mutants at rates much higher than would be predicted by a gradual mechanism alone. And this was work done that recently went into press, and Nadia, a former graduate student in the lab, did a lot of this work. And so this was the first experiment we did that showed that structural alterations can arise in the genome, maybe in a mechanism other than gradualism. We tested the same thing for aneuploidies, for the loss of entire chromosomes, using a similar counter-selectable assay. And again, we found that cells harboring two aneuploidies arose at rates far more frequent than we would have predicted, and actually way higher than even the LOH assays had shown. So many, even 1,000-fold higher rates. And I wanted to note, I forgot on the last slide, these cells are completely wild type, and they're grown in completely normal conditions. They're not exposed to any kind of stressful condition or mutagenic drugs of any kind. So this is happening in just a natively growing cell. So from these quantitative assays, we could come to some early conclusions already. One is that conventional gradual models don't explain the high rates that we were recovering clones harboring multiple structural mutations. And I didn't include this in the talk today, but I'm more than happy to talk to anybody. We also tested and found that these cells harboring two structural alterations are not mutators. They didn't acquire the second mutation because they had gained a primary destabilizing mutation. And I'm happy to talk about this during the question section. But ultimately, these quantitative assays pointed actually to this punctuated burst model, and it suggested that multiple structural mutations can be acquired in a related episode. We sequenced the whole genomes of over 300 clones that had been selected for loss of heterozygosity or for whole chromosome loss. And we were really surprised to see that besides the selected mutations that we had recovered these clones based on, many genomes harbored additional unselected mutations that had co-occurred with the selected one. And so I'm summarizing that here. This is just a schematic showing the 16 chromosomes of the yeast genome. These are diploid cells, so imagine each one of these is a pair of homologs. And what I'm showing you are 4 representative examples of cells that we recovered that had been selected for a primary loss of heterozygosity event or 2 loss of heterozygosity events. And what the yellow boxes are showing here are other chromosome pairs that were affected by unselected loss of heterozygosity. And overall, from our study of LOH, over 15% of clones selected for a single loss of heterozygosity event harbored additional loss of heterozygosity events elsewhere. Aneuploidy was even more noticeable in that 44% of the clones that we selected for a primary aneuploidy event contained additional aneuploidies elsewhere. And for the next couple slides, I'm going to focus and zoom in a little bit here on what the genomes of these aneuploid cells look like, because it's truly, we really think it's quite remarkable. What I'm showing here is just how we represent the yeast genome in a graphical way. And so each set of boxes here is a chromosome 1 through 16. And then here I'm showing you yellow is one homolog from the parent and the black bar is the corresponding homolog from the other parent. So together there's 2 chromosomes. And when we sequenced the genomes of aneuploid clones, we saw that there were that a lot of clones had just the selected mutation that we had seen, and or related or some kind of aneuploidy related to the selected chromosome. And then there were clones that had just a few. But as often as we saw these relatively minor aneuploidization events, we recovered clones that had systemic aneuploidization of the genome that had arisen during these transient episodes of instability. And we've been referring to this in our lab as systemic genomic instability, or SGI. So a type of instability that doesn't affect just a single locus in the genome, but actually affects the whole genome simultaneously. And all of the clones that we sequenced were unique. We never saw the same aneuploidies co-occurring together. And actually each of these genomes is unique. And especially these ones down at the bottom, this is a remarkable amount of aneuploidy to arise in just a short period of time. And this is actually one of our best ideas, arguing that these are probably all arising in a single very bad cell division. Okay, so I want to conclude with kind of what we know now, and then I want to spend a couple of minutes talking about what we're starting to consider next. Overall, our results suggest that cells can actually develop these intricate karyotypic variations really quickly during these short-lived bursts of systemic genomic instability. So during these bursts of instability, which are transient, cells can get a lot of structural variations like aneuploidies or LOH all at the same time. And this begs the question, what would cause bursts of SGI in the cell, especially in a normal cell that's not harboring any deleterious mutations yet? And what we've considered a lot over the last couple years and months is that most genome integrity pathways that maintain the integrity of the genome act systemically. So they act on the whole genome. And probably one of the best examples of this is is mitosis. So the segregation of sister chromatids during cell division. And this is a video of a PTK1 cell, a mammal cell, dividing, and you can see the chromosomes here are being pulled towards the metaphase plate. And it isn't until after every chromosome is aligned on that plate that the cell initiates anaphase. And this highly regulated process is global in that a single spindle is facilitating the division of all of this. And then the mitotic checkpoint is acting as a surveillance pathway to make sure that cells don't initiate anaphase until after they're all ready, until all the chromosomes are aligned on the plate. And so because these pathways act systemically, it follows that perturbation of these very pathways could have systemic consequences. And this is again, very visually acceptable when you consider mitosis, the formation of multipolar spindles or checkpoint dysfunctions alone aren't just going to affect a single chromosome in the genome. They're actually going to affect all chromosomes with equal probabilities. And so we're starting to think that it's the failure of the very pathways that ensure the stability of the genome that are transiently failing to give rise to these really remarkably complex cells. And it turns out that these pathways are very vulnerable to just stochastic perturbation caused by variable gene expression. A lot of the positive regulators and a lot of the really key factors that regulate this mitotic checkpoint, the DNA damage checkpoint, are expressed at exceedingly low levels in the cell. And so any noisy gene expression that could just happen, these are the proteins and these are the pathways that stand to fluctuate the most. And this has been described for a very long time in terms of not a true genetic mutator, which I talked about earlier, but actually as this concept of a transient mutator, which is a genetically normal cell that experiences episodes of instability just due to stochastic noise in the expression of a genome integrity factor or a pathway. And we think this is really intriguing because we know that our cells from our studies are wild type, and that they do go back to a state of relative genome stability, and they carry these new genomes for a very long time. And we're really investigating how long they can carry some of these karyotypic burdens before they become unstable, if they do at all. But Nadia, the graduate student who was in our lab, went on to work on this in her postdoc. And this is a paper that she put on bioRxiv just a couple weeks ago. But basically, she used E. coli to look at the stochastic expression of important proteins in bacteria. And what I'm showing you here is a video of dividing E. coli cells that are expressing REC-A-GFP, which is the homolog for Rad51, an important protein for recognizing double-strand breaks and facilitating double-strand break repair and homologous recombination. And what she saw is that in normal dividing cells, the expression of REC-A or Rad51 fluctuates and actually has a burst-like expression pattern where for some reason cells will just overexpress for a little bit, but these are transient and short-lived episodes of overexpression and underexpression. And so we're operating and starting to investigate a model in which stochastic errors in expression of genome integrity factors.
You're 3 minutes over time. So
oh no, okay, all right. That was the last slide. But basically, I think what we think is happening is that cells may actually just transiently overexpress a protein and they get a lot of mutations at that time, and then they go back to expressing at wild type levels. So yeah, with that, I would like to thank everyone in the Arguesa lab, Lucas and Nadia especially. I'd like to thank our funding sources, Cancer Center and Colorado State, and please feel free to email with any questions.
Thank you. That was such a beautiful talk. You do have a couple of questions, so we start with Patricia.
Okay, I'll try to make it quick, but it's kind of a crazy one in that I'm trying to connect your talk to Dr. Borja's talk. So my question is, you showed an unexpectedly high frequency of LOH and aneuploidies in a lab strain of yeast, right? So if you do that experiment, my understanding is that Cerevisia has some substrains and natural variety, right? Maybe from different beers or whatever.
Yeah.
But if you do that experiment in a bunch of different strains, how much does that frequency vary? And do you think that reflects like human variation that could happen? Yeah.
So that's a great question. And actually, we did these experiments in 3 or 4 different genetic backgrounds. And we saw the same frequencies of these mutations. These are really fundamental processes. And I think that there probably is some subtle variation, but our fluctuation analysis didn't really allow us to pick up on that. But I think that's a great idea. We tried to mimic a lot of the heterozygosity that exists in human genomes. So these strains were not isogenic. They weren't identical. There was a lot of heterozygosity. And I think that that's a really that's definitely something we're working on in the lab now, trying to understand if there's predispositions for bursts, right? But it's a little too soon to say it.
Okay, thanks.
Bob, do you have a question?
Yeah, Lydia, it was a great talk. I really enjoyed it. Yeah, so Hartwell's lab developed a mapping technique for mapping mutations that was dependent on the fact that when a cell loses a chromosome, it's more likely to lose another chromosome. So your results, I mean, take that, miles ahead.
Yeah.
And so it's consistent with that. But they used microtubule poison to do it. They didn't look under normal conditions.
Yep.
So your results suggest, maybe under normal division, there's some sort of problem with the spindle, and then boom, it's like an explosion.
Yeah, that's what we think is happening. And yeah, like you said, the disparate and coincident accumulation of mutations has been reported for over 60 years. It's just, we don't think, without the advantage of whole genome sequencing and being able to look at the whole genome, it was very hard to tell what was, like, how extensive bursts really were.
Thank you. We have the last question from Julie.
Thank you. That was a terrific talk. I guess I had a couple of questions. Did it look like chromosome 15 and 16 had a lower probability of being involved in these mutations?
No, when we actually counted through all the unselected events that occurred, there was no bias towards any chromosome at all.
I see. It looked okay. And I guess if you look at the position along a chromosome, so all the chromosomes have maybe then equal probability. Is there any sort of correlation between, being near a telomere, near a centromere, far from either?
For the loss of heterozygosity studies?
Yes.
Yeah, we looked, and I've gone through that data many times because I just would love to see something there. But we actually saw, we saw a lot of terminal LOH events, but we also saw segmental LOH as well. And there was no bias in those terminal events for telomere proximal sites or centromere proximal sites.
I see. And I mean, have you done, or has anybody done haploinsufficiency kind of screens for genome instability?
Oh, yeah.
Like if so, you're saying that these stochastic changes in gene expression, like, are there some genes you know of whose absolute gene products whose concentrations are so important? Or
yeah, and I think this is a lot like a kind of a Goldilocks dilemma, where you can have too much of a protein or too little. With regard to diploid genomes, I think it's likely, and there's more evidence showing that overexpression of some of these proteins can drive it. But we know really well that if you start messing with the expression levels of MEK and TEL, so human ATR and ATM, you can essentially recapitulate a lot of these phenotypes very quickly. And Tom Pettis at Duke, we've been talking to him quite a lot about that.
Cool.
Excellent. Thank you. Round of applause.
Sorry for going late.
And so the next speaker is Jyoti Mahadevan, postdoctoral fellow at CU Boulder, going to talk about understanding the dynamics of endogenous PARP1 using live cell microscopy. Go ahead, Jyoti.
Can you all see my screen now?
Yep.
Okay. Firstly, I would like to thank the organizers for giving me a chance today to talk at this retreat. And today I'll be talking to you about the work I've done in understanding the dynamics of endogenous PARP1 and PARP2 using live cell microscopy. So poly-ADP ribose polymerases, or PARP1 and PARP2, are two nuclear DNA damage dependent PARP enzymes. There are 17 total PARP enzymes, and PARP1 and PARP2 are the ones that are nuclear and DNA damage dependent. These enzymes utilize NAD+ and put together long chains of poly-ADP ribose on themselves and on other acceptor proteins present in the nucleus. PARP1 especially has a number of functions in the cells. PARP1 is involved in the inflammation response. It is involved in transcriptional regulation. It also acts as a chromatin architectural protein. And most importantly, it functions in DNA repair, both in single-stranded and double-strand break repair. So today I will be focusing on PARP's role as a chromatin architectural protein and PARP's role as a DNA repair protein in cells. So PARP1 in its steady state, it binds and compacts chromatin, and in the presence of DNA damage, it uses NAD+ and activates itself and other nuclear acceptor proteins. So the activated PARP or the PARylated PARP1 has very less affinity to chromatin, and it results in relaxation of chromatin structure. And then this allows the DNA repair machinery and chromatin remodelers to come in and destabilize the nucleosomes. The PARylated PARP1 then acts as a histone chaperone and captures these evicted histones and aids in the process of chromatin assembly in the wake of DNA damage repair. And at the end, there are enzymes called glycohydrolases that rapidly remove these PAR chains, and PARP1 thus resumes its role as a chromatin architectural protein. And then the process of activation of PARP1 is extremely fast. Now, PARP inhibitors are chemical compounds that inhibit the activation of PARP. These are nicotinamide analogs that competitively inhibit the binding of NAD+ to PARP's active site. And many of these PARP inhibitors are now clinically approved cancer drugs. For example, talazoparib and olaparib have been successfully used in treatment of breast and ovarian cancer. And very recently, that is in May 2020, olaparib was approved for prostate cancer treatment. And another PARP inhibitor that I'll be talking to you about today is veliparib, and it is still in clinical trials. An important thing to note is that PARP inhibitors have similar potency for catalytic inhibition. Another feature of PARP inhibitors, apart from PARP inhibition, is PARP trapping. PARP inhibitors are capable of trapping PARP in an inactive state at DNA lesions. So when you have PARP inhibition, there is also inhibition of autopyrylation, and this results in trapping of PARP on DNA strands. This results in the trapping of PARP in the DNA strands. And then also the pyrylation inhibition causes an inhibition of the base excision repair pathways, and this results in increase in double-stranded breaks in the cells. So these double-stranded breaks in the cells accumulate over several cell cycles. Now, there are two situations over here. There are cells that are proficient in homologous recombination repair that can easily repair this damage, and these cells can survive. However, in certain cancers, there are mutations in one of these HR proteins, such as BRCA1, BRCA2, PALB2, or RAD51. These HR deficient cells are unable to repair these DSBs, and this results in cell death, and which is a favorable outcome for cancer treatment. And there's a lot of focus in the DNA repair field for identifying more of these synthetic lethality proteins that participate in synthetic lethality. So another thing to note is that PARP inhibitors, though they don't vary in their PARP inhibition potency, they vary a lot in the PARP trapping potency. And there are semi-quantitative studies that have been performed that show that talazoparib is a more potent trapper than olaparib, which is more potent trapper than veliparib. So the focus of my talk today is going to be about my work in quantitating the accumulation of PARPs at sites of DNA damage, and then how I've made some progress in understanding the dynamics of endogenous PARPs in live cells. And this will be followed by a little discussion on how I figured out a way to quantitate the potency of PARP trapping of various PARP inhibitors. So in order to study the accumulation of PARP1 to sites of DNA damage, I use a method called as laser microradiation. This method uses 405 nanometer laser to induce DNA damage at predetermined regions of interest, such as this red box over here. And then we monitor the recruitment of PARP1 over time. So this is a HeLa nucleus that is going to get irradiated, and this is how the movie looks like. You see a rapid accumulation of PARP1 at sites of DNA damage. So for a very long time, there was no real good method to analyze data that comes out of laser microradiation. So analysis was confined to coming up with average curves like these, and then looking at the T1/2 values, which is the time of half maximal accumulation. The T1/2 value does not provide any information regarding the mechanism of transport, nor does it give you information about the amplitude or the diffusion coefficients. And it also does not account for the variation in the size and shape of the nuclei. So in order to help with this, we came up with a mechanistic way of quantitating the fluorescence accumulation after DNA damage. So in this method, we process each nuclei separately, individually, and we first create a nuclear mask of the said nucleus, and then simulate the accumulation of these proteins by simple free diffusion. And then we match the simulated curve with the experimental curve and come up with values for the D effective, which is the effective diffusion coefficient, and the fraction of mobile fluorescent protein. So a big advancement over previous techniques is that here we consider the shape and size of the nucleus, and because D effective is a value, it can be compared against other proteins, against the D effective of other proteins, and one can learn a lot of things using this particular method. We have also been recently successful in automating this particular method, and we have put it on a freely available platform, and this will be available for all the researchers studying their favorite protein, and they can do similar analysis. And then the next topic I'll be talking to you about is my work in understanding the dynamics of endogenous PARPs in live cells. So far we've been working only on overexpressed proteins or transiently expressed proteins. So we thought that we would look at this in a further little more detail. And for this, we used CRISPR-Cas9 based genome editing tools to introduce a 3x flag halo tag on the endogenous PARP locus. And this result, we can see that we have a robust expression of halo PARP1 and halo PARP2 in our U2S cells. So in order to understand the accumulation kinetics of halo PARP1 and halo PARP2, first we stain these cells with JF646, which is a Janelia floor dye. It's a halo tag ligand, and we perform laser microradiation assay followed by QFAT analysis. So what you can see here in the first movie on the left is that there is a rapid accumulation of halo PARP1 to the sites of DNA damage. And on the right, halo PARP2 is much slower compared to that of halo PARP1. And then when we analyzed a bunch of different nuclei, we found that halopart 1 accumulates 6 times faster than halopart 2 at DNA lesions. Now we took this a step further and we wanted to understand the behavior of individual molecules of halopart 1 in live cells in the undamaged condition. So here we stain the cells with a low nanomolar concentration of the halotag ligand JF646 so that we can visualize individual molecules of part 1. We also do high-low microscopy where we direct the laser light at an angle through the sample so that we get a Z of 10 nanometers. And we acquire these images at an extremely fast rate, which is 100 frames per second. So here is a movie you can watch of halopart 1 traversing the interior of the nucleus in the absence of DNA damage. And we collect a bunch of such movies and process them, and we fit a model, which is a 3-state model, to our data. And I've represented my data in the form of histograms like these, and I'll tell you what these histograms are. So these histograms depict the distribution of all of the particles in the first 7 frames of the movie. The x-axis represents the displacement in micrometers, and the y-axis is the probability of the particle that starts at the origin and it arrives at a particular location in the time delta t, which is either 10, 20, or 30 seconds, and so on. So if you notice carefully, there is a dotted line over here that indicates the fit of our model. And this suggests that we get excellent fits of these movies to the 3-state model. And we can learn a lot of things from movies like this. For example, we get values for the diffusion coefficient of the bound particles, the slow moving particles, and the fast particles. And what we can find over here is that the diffusion coefficient of the fast moving particles is more or less similar in both halopart 1 and halopart 2. What is different here is the fraction of bound particles. We see that there is a higher fraction of bound particles for halopart 1 as compared to that of halopart 2. So what we get from these experiments is that we identify 3 different populations of part 1 and part 2 in live cells, and a higher fraction of part 1 remains chromatin bound when compared to that of part 2. So we have extended our efforts to understand part 1 dynamics upon induction of DNA damage. This is largely work in progress right now, but we are using a novel approach that we developed in the Luger lab, where we use the laser microradiation assay in conjunction with the single molecule tracking method. So this particular assay gives us the power to ask questions about the kinetic behavior of PARPs both inside and outside of the damaged region within the live cells, and understand the biology of PARPs in further detail. So here is one of these U2OS nuclei. You can see the accumulation of halopart 1 particles upon the induction of DNA damage. So I would like to focus your attention between 20 seconds and 30 seconds of what you see over here. And you can see particles slowly trickling in and coming to the sites of DNA damage. So there you go. So that was that. And then the last part is how we figured out about quantitating the potency of PARP trapping of various PARP inhibitors. So as I've told you a bit earlier, The potency of PARP inhibition remains the same for mostly for all the inhibitors, but the potency for PARP trapping is much different. So here, and it is very important to quantitate the potency of PARP trapping for studying new PARP inhibitors in the field. So in order to do this, we quantitate the PARP trapping using the cell lines that I've developed, and we use endogenous PARP. We look at endogenous PARP1 trapping, and here we use the laser microradiation followed by live cell imaging. But instead of studying the accumulation kinetics, we look at the off rate of PARP1, that is the rate at which PARP1 exits the damaged sites. So our result looks somewhat like this, and we use a single exponential model to study the off rate of PARP1. So what we expect over here is that because PARP1 is trapped at damaged sites, it takes longer to release from the damaged sites, and this would cause a decrease in the off rate of PARP1. And we looked at 3 different inhibitors, olaparib. What you can see here is as we increase the concentration of inhibitors, the olaparib concentration from 0.6 to 20 micromolar, you can see a drop in the off rate of PARP1, which signifies that there is increased PARP trapping with increased concentration of the inhibitor. Similar results can be obtained, found also for talazoparib. And here, what is important to note is that you need a much lower concentration of talazoparib to cause PARP trapping. And then we also looked at veliparib, which has not been in the clinic yet, and we do not see a significant drop in the PARP off rate over here. So basically, it's not a very good trapper. So talazoparib is a most potent trapper, whereas the veliparib is a poor trapper. So the take-home message that I have for you today is that we have identified 3 populations of PARP1 and PARP2 in living U2OS cells. And then PARP1 and PARP2 sample nucleus space with similar kinetics in the absence of DNA damage. And obviously, a higher fraction of PARP1 remains bound to chromatin when there is no DNA damage. And we have developed a method to evaluate the potency of PARP inhibitors using live cell imaging. And this can be used for any new inhibitors that are going to come into the field. So with that, I would like to acknowledge my lab for the constant support, especially Carolyn for all the mentorship and all the reagents from Luke Lavis's lab in HHMI, who gave us an endless supply of the JF646 ligand, and our collaborators and all the funding that we have. Thank you.
Thank you. That was great. Are there questions for Jyoti? I'll start off with one. You described the kinetics without DNA damage. Mm-hmm. Do you think that they will be different with DNA damage? And if so, how might they be different?
I think we have looked at this with our bulk assay, the laser microradiation assay, and we have seen that PARP1 is, when they come to sites of DNA damage, PARP1 is 6 times faster than that of PARP2. And we expect to see something similar, but we also have to understand that we are looking at individual molecules now, and the things that we would discover here would be maybe different as compared to what we see in an overexpressed system and in a bulk process. So, I mean, I cannot really tell you what I expect, but I think PARP1 will be still faster than that of PARP2.
Thank you. Other questions? Oh, yes, Julie, you have a question?
Hi, that was really cool.
Thank you.
I'm just wondering if you've looked at PARP dynamics in a cell that's not a U2OS cell, just because, U2S being alt have, the ATRX mutations, and, that one could imagine. I'm just curious of whether you have done these experiments in another cell line to see if PARP dynamics are different.
Yeah, I mean, I have not made endogenous CRISPR-based clone in another cell line. I have done similar experiments looking at Halo PARP1 in an overexpressed system in HeLa cells. I don't think I collected so much data and evaluated it, but the numbers look pretty similar to what I have shown today in HeLa cells as well. But I think after I listened to your talk, I think that thing came into my mind that yes, these are all cells. I've noted that point.
Okay, thank you.
Thank you.
Okay, thank you. Another round of applause for Jyoti. There we go.
Thank you.
All right, so the last two talks of the retreat are by assistant professors who are also mentor members of MCO. Joshua Black from the Department of Pharmacology is going to talk about a conserved DNA re-replication response in adaptation to environmental stress. Josh, go ahead.
Yep. Everyone see my slides?
Yes.
Excellent. So I'd like to thank the organizers for the opportunity to talk today. My lab is interested in understanding DNA re-replication, and in particular, a process by which cells can employ this to change the copy number of specific regions of DNA in response to the environmental cues and how this goes awry in cancer. So we call this process transient site-specific gains, or TSSGs. And a lot of background from my postdoctoral work and what my lab has done, we found that cells can increase copy numbers. So there's a DNA fish that shows an extra copy of this red region that actually disappears during cell cycle. This can be induced by environmental factors like hypoxia, that if you then restore cells to normal oxygen, you remove the extra copy. It's an epigenetically regulated process using primarily methyltransferases and demethylases so far, though we think other epigenetic pathways are important. It can be regulated by microRNA regulators of those epigenetic enzymes, as well as inhibitors or cofactors of those enzymes as well. And so this is a druggable process that we hope to learn more about so that we can control this from a cancer perspective. These TSSGs are derived from DNA re-replication. They occur only in S phase, and they're lost by the end of G2. So this means that this extra copy of DNA is not present for the entire cell cycle, and it's not passed on and inherited by the daughter cells. It actually disappears before nuclear envelope breakdown. This re-replication generally comes from inappropriately open chromatin states, so you can target specific regions of the genome and open them inappropriately, or directed by signaling pathways in order to do that. If you look at regions that are frequently amplified in tumors, they also undergo transient site-specific gain in cellular models, and you can actually use amplification data from tumors to predict regions that might do this. So this implies that the TSSGs could be precursors to these inherited copy number alterations, and that's what I'm going to talk about today. They can be driven by environmental conditions such as hypoxia and also metal exposure, which is what I'll be talking about today. And as far as we know so far, these are conserved across species, and we haven't observed any cell type specificity. So whatever cell types we expose to the given environmental condition or the epigenetic deregulation will generate the transient site-specific gains, regardless of whether or not the region is expressed. So the major questions that we're looking to address are these transient site-specific gains precursors to inherited gene amplifications? And what biological processes can drive this? Like, is this a regulated biological response or just something that goes wrong in cancer cells? And so we elected to use a classical model of gene amplification, the metallothionein genes, to address both of these questions. So the metallothionein genes are a group of metal responsive genes that are located in human cells on chromosome 16. There's at least 14 genes and a few pseudogenes, and they're all in the same locus that extends about 150 kilobases in the middle of chromosome 16. These are long known in history to be amplified from heavy metal exposure. So this was originally done in the late 70s. And if you select cells for cadmium resistance, they get large scale amplifications of these entire loci. This was really done in rodent cells and hasn't really been confirmed in human cells. But the genes are also evolutionarily prone to gene duplication. So in yeast, there's 1 metallothionein. In mouse, there's 4. And in human, there's 16. So there's a wide spectrum across different evolutionary timescales. These genes normally provide protection against heavy metal toxicity, so they bind to metals with high affinity, but they also regulate homeostasis of essential metals like copper and zinc. And so you can imagine they control lots of processes of every transcription factor enzyme that uses those metals. Interestingly, the metal we're used for our model is cadmium. Cadmium's been shown to transform normal breast cancer cells and is elevated in tumors compared to adjacent normal tissue, regardless of whether patients have actually been exposed to cadmium. So it somehow accumulates in the breast tissue. These metallothionein genes also play a role in tumor drug resistance. They obviously can bind all the metal-based chemotherapeutics like cisplatin and help to clear them or diminish their effectiveness. But they also provide resistance to other therapeutics, such as microtubule inhibitors, irradiation, all through controlling the different availabilities of metals and the function of transcription factors and even p53 within the cells. So we recapitulated some of these old experiments from the 70s in human cells. We used NDA NB231 breast cancer cells as a model. Our parental cells, luckily, were diploid for this locus, even though the cells themselves have many other chromosomal alterations in them. They are diploid for chromosome 16. We developed FISH probes for the chromosome 16 centromere and the metallothionein locus, which is in red. And then we exposed cells to metals and gradually raised the concentration over a long period of time until the cells were resistant to really high levels of cadmium, up to 100 micromolar. And what we observed is that early on, at sort of 5 or 10 micromolar cadmium, we observed one extra copy of metallothionein in about 10 to 15% of the cells. And as you maintain these cells in cadmium and increase the levels, you get a gradually increasing amplification of the metallothionein genes. But you also start seeing an increase in amplification of the 16 centromere probe, which is about 10 to 15 megabases upstream on chromosome 16. And so this suggests that we're getting sort of two different stages of amplifications: an initial focal gain that may or may not be extra chromosomal or transient, as well as eventual arm level or perhaps whole chromosome gains the longer you expose these cells to cadmium. So Greg Wright, who developed the system, went on to ask, are any of these gains transient? Are they our TSS genes? The way we address this is our TSS genes are only present during S phase. So if we arrest the cell cycle, we should lose the extra copies of the region if they're transient site-specific gains. So we do that with hydroxyurea, which arrests the G1S. What we found is that in the cadmium-resistant cells, we actually lost some of the focal gains and kept many of the arm level gains. And so we measured this by FISH, where we lost about 30% of the extra chromosome cells with extra chromosomes, or by genomic qPCR, where you lose about 30% of the total DNA content for the metallothionein locus. So at least some of our gains in these cells are transient. If you do this at the early stages at 5 or 10 micromolar cadmium, you actually lose all of the extra gains. So they start off entirely as transient in the cells. So we've developed a way to look at re-replicated DNA in the lab that we called re-rep-seq. We published this year if you're interested in seeing about it, or talk to me later about how we could use this technique in other systems. So this allows us to sequence specifically re-replicated DNA in cells. And so Johannes, who developed the technique, applied it to our cadmium-resistant cells. And we found on chromosome 16 a large region that has strongly increased signal in the re-replication sequencing compared to our parental 231 cells. I've coupled this with replication domain sequencing here that shows that this region actually spans multiple replication domains of both early and late timing. And so it's not clear what's restricting or how this works. I should also point out that this is entirely population-based. So we don't know that every cell re-replicates the whole region. There's obviously some domains that are stronger than others. The metallothioneins are represented in this large hump. And this was also the only strong region we found throughout the entire genome on the cell. So almost all the other regions, all the rest of the chromosomes are pretty much the same between parental and cadmium-treated cells. So I identified in here sort of a minimal region of re-replication that's about 5 megabases in length. And we want to know if this is something that's reflected in metallothionein amplifications in tumors. And so what we did is we went to the TCGA patient copy number data. And we found about 100 patients out of the 1,300 or so breast cancer patients in TCGA that had metallothionein gene amplifications. So the metallothionein locus is here in this black box. And there's many different degrees and sizes of amplifications in this region. And so what we did is we thought that if the re-replicated region might be a basis for some of these potentially inherited copy number changes, that the breakpoints of amplification in the patient data should be overrepresented compared to other regions of amplification in those same samples. And so what we did is quantify the number of breakpoints that occurred within the re-replication domain. And then we randomly sampled the rest of the genome for domains of equal size and asked what's the frequency of breakpoints in those domains. And what we found is that the metallothionein region was highly enriched compared to most of the rest of the randomly sampled domains and was in the top 3% of all regions genome-wide for breakpoints. So this suggests that maybe these re-replication domains might be precursors to some of the inherited amplifications we see in cancer and might be precursors to inherited amplifications in general. So to summarize what I've told you so far is that the MT locus undergoes DNA re-replication and forms these TSSGs in response to metal exposure. TSSG formation precedes a stable inherited gene amplification in the cells. What I didn't show you for lack of time, but I'll tell you is that this is an epigenetically regulated process that's controlled by EZH2 and PRC2, which are the histone 3 lysine 27 methyltransferases. You can recapitulate the metal results simply by inhibiting EZH2 long-term. So if you inhibit EZH2 in cells for 30 passages, they will also integrate and inherit these copy gains, and this will become important going forward. So you don't need the selective pressure of the cadmium or the metals to generate these copy number changes. So our next question was, how do these metallothionein TSSGs transition to inherited gene amplifications? So we got a hint at this from doing some metaphase spread analysis of the cells. What we found is that our cadmium-resistant cells had high levels of tandem duplications, both including and not including the centromere, and in some cases formed repeats down entire lengths of chromosomes. So this suggested that one of the most likely pathways here is that this is going to be a homologous recombination dependent pathway generating these tandem repeat analyses. So to investigate this, we went back to our EZH2 inhibition system. So this is a population analysis by fish of copy number. And so the more each dot represents one cell, the further you go up the graph, the more copies you have of both the centromere and the metallothionein. So what we found is that when you inhibit EZH2, at around passage 30 to 35, you start accumulating these chromosomal gains, either arm level or higher, that are inherited. And this gets worse the longer you leave them in EZH2. However, when you put them in an HR inhibitor at the same time and do the same cultures, you don't really accumulate these gains. And so what you can see here is that you lose these higher levels of inherited gains, but you're still generating the extra transient copies. So they're no longer able to inherit the extra copies. So we next, since we did re-replication sequencing, we actually went back and did whole genome sequencing of paired end analysis to see if we can find any structural variants that support this idea. And so what we did is we performed a grids analysis on the whole genome sequencing from the cadmium-resistant cells. And we identified 585 variants that originate from the metallothionein locus on chromosome 16 and are distributed to pretty much every other chromosome in the cell. This may look like a completely random distribution pattern. And so we asked, is there anything specific about where these integrations are occurring or where these translocations are lining up? Of the 585 variants we found that originate in the locus, 375 of those had homology to the metallothionein locus. So it's consistent with a homologous recombination model. That homology had an average length in base pairs of about 110 base pairs. If you scramble those sequences or if you randomly sample the genome instead, you have almost no homology length in those same samples. It's a pretty strong homology. This is the E-value of the BLAST analysis. And while there's a wide distribution, it's significantly enriched over both the scramble or the random distribution. But the real interesting thing was that this homology is actually enriched very close to the breakpoint. So we actually sampled homology within 500 base pairs of either direction of the grid's predicted breakpoint. And the homology is highest right at that breakpoint, peaking just before, suggesting that most of these loci have this homology that's very adjacent. So we asked, like, what is this homology? Like, is there something specific to these cells, or is it just random spots in the genome? So we did this. It turns out that it's highly enriched in repeat elements, and they are almost all ALU elements. So of those 375 homologous regions, approximately 280 of them were repeat elements. And the highest enriched class of those are all different families of the ALU elements. So the top 10 of these outside of one simple repeat are all different families of ALU elements. So this suggests that we're getting homologous recombination between re-replicated fragments that contain the ALU elements and other ALU regions in the genome. So the title of my talk mentioned that this is a conserved process. So we reasoned that if this was a real sort of biological response, that different organisms should also be responding to metal exposure the same way. And so it turns out that metallothionein genes are conserved in yeast. The Saccharomyces cerevisiae homolog is called CUP1. This has been used for many years as actually an inducible promoter. If you expose yeast cells to copper, they will turn on your favorite gene of interest. And so people have used this for a long time. It also turns out that you can select yeast in the presence of copper and they will amplify the CUP1 locus. This is total genomic DNA here. If you expose yeast cells to copper, you greatly increase the number of copies of CUP1, but not other surrounding genes. So it's pretty specific. So we performed our re-replication sequencing on these yeast cells that were selected in copper. And we found a high enrichment of the CUP1 locus in the selected populations, but only when labeled for our technique for re-replication sequencing. When we sequenced this analysis, we found a very strong enrichment directly over the CUP1 locus and sort of some neighboring gene regions here that significantly enriched over the control elements. So we asked in the yeast, we also performed whole genome sequencing to analyze what was happening to structural variants in yeast. In yeast, we identified about 600 variants originating from the CUP1 locus on chromosome 8 in yeast. Again, these were distributed to pretty much all other chromosomes. We did not find any strong homology in the yeast, so nothing, none of these 100 base pair, 200 base pair homology linked the regions at the break sites. Instead, we found a very strong enrichment for microhomology. Almost 90% of the regions had a microhomology to the CUP1 locus. So the average homology length in base pairs was about 9 base pairs in our samples, which was significantly lower in either scrambled or randomly genomic paired regions. These were enriched almost in the exact same spot in all of the samples, about 5 base pairs downstream of the breakpoint. And so this is very consistent with what's known about microhomology mediated repair in cerevisiae, is that there's a very close homologous region immediately downstream of breakpoints. So in both yeast and human cells, we're using a homologous recombination method to transition from a DNA re-replication transient event into a long-term stable inherited gene amplification. So in summary, I've told you that transient gene amplifications through DNA re-replication can precede these inherited events. The homologous recombination is required for that transition from stable gene amplification of the locus. That the region of re-replication of the locus correlates with amplification breakpoints in tumors, suggesting that the origins of those might be from re-replication events. And that this is conserved across species from yeast to human, that these metal-induced or environmental exposures can lead to transient and eventual inherited gene amplifications. So our model from both yeast and human is that as you expose cells to stress for a period of time, you get an initial quick transient response that gives you extra chromosomal transient amplifications of the gene. But if you keep those cells persistently in that stress, eventually you will end up with cells that have reintegrated and expanded and increased the copy number of those genes. And that allows them to better survive long term. And this is through a homologous recombination dependent mechanism. So with that, I'd be happy to take any questions. The work in the lab was done. Greg Wright developed the metallothionein human cell system. Johannes developed the re-replication sequencing. And Phil has done a lot of the computational analysis on the homologies and identifying the repeats and breakpoints of that. And I'll take any questions.
Thank you, Josh. And I was reminded that even though Josh was a mentor member when we were planning the retreat, he was recently promoted to full member. So congratulations.
Oh, thank you.
And you have a question from Haida.
Hey, Josh, that was a great talk. I'm wondering, and I don't know why I never asked you this before, but in the case of the human samples that you were analyzing to look at the metallothionein gene amplifications, was that associated with any alterations in EZH2 or other chromatin modifiers that could explain, other than, you know, being exposed to heavy metal, why you would get that?
Yep, so we've looked at that. There's not a lot of amplifications or deletions of EZH2 in the breast cancer patients. There are frequently mutations, but they're not well characterized. There's lots of mutations that will increase function of EZH2 and others that will decrease function. And since we only started with 100 samples out of the 113 to begin with, we didn't have nearly enough to do any sort of comparison with breakdowns of EZH2 mutants or not. So that's a great question, and we might predict that in a larger sample, but we haven't done that analysis yet or correlated with EZH2 expression levels. I should point out, though, that there's a lot of clinical development around EZH2 inhibitors for use in several different cancer types. Obviously, we predict that long-term use of those might be detrimental
Right.
in patient populations. But short-term use should theoretically be fine.
Yeah, okay. Yep. Thanks, Josh.
Julie has a question. Go for it.
Josh, that was a really cool story. I just wondered if you could tell us a little more about the mechanism by which the extra copies during TSSG are eliminated during G2.
Yeah, so I use disappear for a reason because we have no idea. So there's sort of three possibilities. The first is they're degraded in the nucleus by nucleases, and there's about 52 of them that are nuclear dependent. They could be exported out of the nucleus and degraded. They could be exported out of the cell. Or there's also the fact that most of our detection is all by DNA fish, which we don't have a way to detect cytoplasmic versions of fragments yet. So they could just be exported but not actually degraded or lost. My favorite model is that they are actually exported out of the nucleus into autophagic vesicles and degraded in the lysosome. I have two weak pieces of evidence for that. The first is that about 3 years ago now, there's evidence that DNA double-strand, pieces of chromatin with DNA double-strand breaks that still have nucleosomes on them get exported out into an autophagic vesicle. So shown by live imaging with GFP tagged histones, for example. The second is that if you take the cancer data and you sort for tumor samples that have a deletion of DNase 2, which is the lysosomal DNase, you see enrichment for amplifications of regions that also undergo TSSG. So this is something we're interested in investigating in the future, but I don't have any experiments to really tell you about for that right now.
Very interesting. Thank you.
James, if you have a real quick question.
Real quick. So why didn't you map any breakpoints back to the same chromosome?
So they're almost all entirely in the same locus. So we just took them off for simplicity, but there are lots of suggestions that it's tandem duplications inside the metallothionein locus as well, and they just string along. And so we left those out for simplicity, but there's tons of those there, both in the yeast and the human.
Thank you. Round of applause for Josh. And the last talk is going to be by Nausicaa Arnold, who is in MCD biology at Boulder. She's going to talk about using CRISPR-Cas9 screens to study DNA repair pathways, role of APAX2 in alternative end joining. Nausicaa.
Yes. So I'm going to try to share my screen first. Okay, can you see it?
Yes.
Yes. Okay, thank you. So, well, it's a pleasure to talk today. I am sorry if I am going to repeat what other people have introduced because I was teaching this morning, so I just joined. But anyway, so my lab is interested in double-strand break repair and in telomeres. And today I'm going to talk about how we are using CRISPR-Cas9 screens to study DNA repair pathway, and especially the alternative end joining pathway. So, as it was probably said multiple times today, one of the hallmarks of cancer cells is the presence of high genomic instability. And a lot of times you get this high genomic instability by having mutations in DNA repair genes. So almost all cancer cells have some DNA repair genes that are mutated, and among them, a lot are within homologous recombination. So double-strand breaks can be repaired by three pathways. You all know non-homologous end joining and homologous recombination, but there's a third pathway called alternative end joining. It's also called MMEJ, or Pol theta mediated end joining. And it's considered a backup pathway for homologous recombination, and it works by using microhomology around the break to repair DNA. It's intrinsically toxic. Okay, so this is how the different double-strand break repair pathways are used at double-ended double-strand breaks. However, these double-strand breaks are very rare naturally in cells because they occur through radiations or when you use Cas9. And actually an overwhelming majority of the double-strand break that occurs in cells occur during replication. So during replication, when there's a single-strand break, the replication fork is going to convert the single-strand break in a very peculiar type of double-strand break called single-ended double-strand break. And these breaks will never be repaired by NHEG that is actively suppressed. Instead, they're repaired by homologous recombination. So when homologous recombination is mutated, which occurs frequently in cancer, then the cell relies solely on alternative enjoining to repair these breaks. This means first that if you also suppress alternative enjoining, then the cells cannot repair these breaks anymore and they die. And this synthetic lethality has been used a lot to treat HR deficient cancer cells. We've heard a talk about PARP inhibitors, and what is now believed is that PARP inhibitors that plays a role in many pathways is also involved in alternative enjoining, and that is what is killing the cells that are deficient for HR. So, however, there's a lot of resistances to PARP inhibitors. So there's a huge effort in the field now to find novel targets for HR deficient cancer cells, and especially novel targets within the alternative enjoining pathway. So a lot of research is being done to set up inhibitors against POLθ, which is the translational polymerase that acts during alternative enjoining. So it's encoded by POLQ gene, and we'll talk a lot about that later. But my lab has decided to actually use this synthetic lethality in CRISPR screens to identify novel alternative enjoining factors and therefore use them, see if they can be used as potential targets down the road. So the concept of such a screen is pretty simple because there's a synthetic lethality between homologous recombination and alternative enjoining. If you infect HR positive cells with a genome-wide guide RNA library, all the guide RNAs that target alternative enjoining are not going to be lethal in cells that have HR, but they are going to be lethal in cells that do not have HR. So the way we did that, and there's a lot of labs who have done synthetic lethality screens against BRCA1 or against BRCA2, but we were really interested in knowing what is synthetic lethal with generally all the HR pathway. So to do that, we needed isogenic cells that were either HR proficient or HR deficient, but in different steps of the HR pathway. So we use HT1080 cells that are HR positive cells and depleted by CRISPR-Cas9 either BRCA1 that is quite upstream in the pathway or PALB2 that is rather downstream in the pathway. And we selected a single clone, checked that this clone was each time that the knockout was good, and then used this clonal population in the screen. So in the screen, we took the parental HT1080, the BRCA1 knockout, the PALB2 knockout, infected with a CRISPR-Cas9 genome-wide knockout library, and let the cells grow for 10 population doublings. Then we compared the guide RNA found in BRCA1 knockout to the wild type, and this is the result here, and you see that there's a lot of genes, including a lot of genes that belong to the Fanconi anemia pathway. And this is the PALB2 result. And you'll notice that the set of genes that we have is quite different each time because each gene has its own factors that are synthetically lethal with it. So again, what we wanted to do was to see what is synthetically lethal with generally the whole HR pathway. So then we plotted here the BRCA1 screen. So this is the p-value for the BRCA1 cells versus wild type. And this is the p-value for the PALB2 versus wild type. And in that condition, we see much less genes that are significantly depleted in both BRCA1 and PALB2. And remember that we wanted to do this screen to identify alternative enjoining factors. And we were very happy to see that all the alternative enjoining factors that are known so far were significantly depleted in our screen. So here in blue. Above this, we have three other genes. CYP2A, which is a cell cycle regulator on DNA damage. ALC1, which is also a chromatin remodeler involved in DNA repair, and AP2, which is an AP endoexonuclease that is involved in base excision repair. So we wanted to test whether these proteins have a direct function also in alternative unjoining. The way people do that generally is to use DNA repair reporters. So you introduce a plasmid that you can cut with an ISC1, and the way the plasmid function, the way the reporter function in this case here, if it repairs through alternative unjoining, meaning if you use microhomologies to do the repair, it is going to express GFP. So unfortunately, these reporters are really not perfect, and we're not very happy with the results. So first, we're doing a second reporter that is supposed to work better. And secondly, we are going to, we used another way to look at alternative unjoining using telomere fusions. So how can you use telomeres to look at alternative unjoining? Well, telomeres, because they're at the end of chromosome, they look like double-strand breaks. So they evolve this complex network of proteins that inhibit both the DNA damage signaling pathways and the DNA repair pathway. So you can really play with knocking down some of this protein to activate just the one pathway you're interested in. So that's a very powerful tool to study either non-homologous unjoining or alternative unjoining. So let's see how it works. If you suppress TRF2, which is the main protective factors of telomeres, then you relieve the ATM signaling inhibition and the NHEJ inhibition, and suddenly the chromosome all fuse through telomere-to-telomere fusions, and this fusion occur by NHEJ. So by measuring the amount of fusions, you can measure NHEJ activity. Now, if you do the same thing, but this time you deplete Q70 or Q80, that will lead to two things. First, Q70, Q80 is a main factor of NHEJ. So if you deplete Q70, Q80, you have no more NHEJ. So all the fusions you look at cannot be NHEJ. But Q70, Q80 is also an inhibitor of alternative unjoining. So suddenly you also relieve the inhibition on alternative unjoining, and the fusions you see here are all due to alternative unjoining. So you can use that as a readout for alternative unjoining. There's a little trick though, is that Q70 or Q80 knockout is lethal in human cells, but it's not in mouse cells. So we had to turn to mouse embryonic fibroblasts. So we took MEF cells that have an inducible TRF2 knockout. And so in that case, there's Q positive cells. So what we're looking at is NHEJ fusions. So if you don't add tamoxifen, you don't suppress TRF2, you have very few fusions. If you now add the tamoxifen, you have this huge increase in fusion, and this is dependent on NHEJ because this is sensitive to an NHEJ inhibitor. So we then did a Q80 knockout, and in these cells, you have fusions that no longer are sensitive to an NHEJ inhibitor, but instead are sensitive to an alternative unjoining inhibitor. And now we can take these cells and add guide RNAs to knock out our candidate genes. So first, non-targeting RNA, we got the same result. POLQ is our positive control. It's involved, it's absolutely required for alternative unjoining. And you see that the suppression is very clear. We were very excited to see that APE2, APEX, is also absolutely required for alternative unjoining. If you don't have APEX, you have no fusion through alternative unjoining. And then CYP2A and ALC1 don't seem to be involved in alternative unjoining. There's no decrease of the fusions here. So the first conclusion is we identified APE2, which was known to be a base excision repair protein, as also playing a critical role in alternative unjoining. So the next question were which domains of APE2 are involved and what is the mechanism of action? So APE2 has different domains. The endonuclease and the 3', 5' exonuclease domain here, the PCNA interacting domain, or PIP, and a zinc finger DNA binding domain. And it was shown that for base excision repair, all of these domains participate. So the endo and the exonuclease are required for base excision repair, and the PIP and the zinc finger domain promotes these exonuclease activities. So are these domains also necessary for alternative enjoyment? So we built mouse AP2 construct that are either wild type, exo-dead, endo-dead, no PIP domain, and a mutation in the zinc finger domain that disrupt the DNA binding. And we complemented with this construct our MEF-Q80 knockout that also have an AP2 knockout. And as you can see, we found that if you have a knockout in either the exonuclease or the endonuclease domain, you're not able to rescue the loss of AP2. You can't make any chromosome fusion through alternative enjoining. But the PIP domain and the zinc finger domain don't seem to play any role here. So now, what is the mechanism? And unfortunately, I don't have the answer, but we do have a very strong hypothesis and a very well way to test it. Everything is ready. We just have, we don't have the result yet. So the way alternative enjoining works is depicted here. So you first have resection, the 5' 3' resection that leaves this microhomology open to annealing. There's annealing of this microhomology, and when there's annealing, there's formation of these three prime flaps. Sorry. And then there's degradation of these flaps, and then POL-Q is going to polymerize, and XRC1 ligase 3 is going to ligate the end. However, we don't know what is removing these flaps. And because AP2 is a 3', 5' exonuclease and endonuclease, it is the perfect candidate to remove these flaps. How to test it though? Well, what we thought is if AP2 is absolutely required to remove these flaps, then it should be dispensable when you don't have the flaps. And you don't have the flaps if the microhomologies are directly flanking the break site. So in that case, upon resection and annealing of the microhomologies, there's no flaps here. And we think that in that case, AP2 is going to be dispensable for alternative enjoining. Now, to test that, we made a very specific DNA repair reporter using, so this time, there's an M-cherry cassette which is interacted with these microhomologies here, and it can be cut through expression of Cas9 and the guide RNA here. And in this case, there is going to be formation of flaps, which, as we think, are going to need AP2 to be degraded. We have a second reporter where, however, the microhomologies are just next to each other. And so when Cas9 and the guide RNA are going to cut, this will lead upon annealing of these microhomologies to no flap. So if our hypothesis is true, we should see M-cherry fluorescence upon Cas9 expression with both of the reporters in wild type cells, none of those with a POL-Q knockout. But if we do an Apex2 knockout, we should not be able to see alternative enjoining when there is flaps, but we should see it when there is no flaps. So to summarize, we've seen that AP2 is part of the alternative enjoining pathway, and we believe it's removing the flaps that form upon annealing of the microhomologies. This is important because, well, first, we get a better comprehension of the alternative enjoining mechanism, which is so far quite, poorly understood compared to other pathways. Also, AP2 is likely to be a very good target for an inhibitor to use against HR deficient cancer. And this also reveals the fact that in DNA repair, a lot of proteins are not just working in one pathway, but they have plenty of roles in different pathways. And my lab is also trying to look at the redundancy and identify through genetic interactions the different function of different genes. And for that, we are performing a large combinatorial CRISPR screen to map all the genetic interactions between DNA repair genes. So with that, I would like to thank everybody who did that work. So Hubert Fleury is a very talented postdoc in my lab, and he did most of this work. But this is really a joint effort of almost all the lab members, including the undergrads, who have been counting chromosome fusions during all summer. So I would like to thank everybody, and I will be very happy to take questions. No.
Great. Thank you.
Stop sharing. I should have stopped sharing. I'm gonna share again. Sorry.
Julie has a question. Go ahead.
Yes.
Hi, Nausicaa. That was great. That was great. I maybe you said it and I didn't catch it. Can you separate the endo and exonuclease domain activities mutationally in APE2?
So we have mutants. We have one mutant that is supposed to be the exo and one that is supposed to be the endo. To be completely honest, I completely trust the exo det because it's an alignment with, so APE2 is very close to APE1 and to the bacterial exo3. And this is really the catalytic site. The endonuclease was published, this mutant was published by Steve Lage in their paper on APE2. They claim it's an endonuclease det, but they don't show that it is one, and they don't reference any other paper. So I'm not so sure, honestly. And also in the same paper, they mutate the PIP domain and their mutation is completely outside the PIP domain. So,
yeah,
so
maybe you guys will separate them.
Yep.
Thanks.
Other questions for Nausicaa?
I have a quick question. I thought that the CTIP and mRNA complex would take care of the end trimming or resection. Why do you still need the APE2 activity?
So the CTIP mRNA, so it's it has not been shown to do that. I mean, it's doing the first resection that leads to these 3' large overhangs, but it is not clear that it's doing this flap trimming. So actually it was proposed that this flap trimming was done by ERCC1 XPF. And at first, ERCC1 XPF was thought to be an alternative and joining repair protein because in vitro it does that job, but in vivo it doesn't. So it's still unclear what does that.
Okay, thanks.
Well, thank you. And another round of applause for Nausicaa. And I think we're ready for concluding remarks by Patricia.
Okay, I'll keep this short because due to the engagement today, we're a little over time. But I really have 2 important points I want to make. One is that we all really appreciate that the keynote speakers and the invited speakers and the speakers selected from abstracts gave really excellent talks today. So final round of applause for them. Here's mine. Okay, now I can't stop it. And then the other point is something I am going to show briefly one slide about, and that is what I think both Craig and James emphasized at the beginning, and that is that one of our purposes today was to get you guys to think about how, this is a very challenging question, how molecular mechanism type work really does affect health disparities and how we can apply our work to health disparities. So I can't quite reach my, let's see, trying to make this a little bit bigger so you can see it. So Craig wanted me to point out, first of all, that there's money for applying your work to health disparities. And I know this is a bit difficult to understand and maybe intangible for some of you. So I thought of a couple things during Dr. Barroyo's talk. And we've been having this discussion for quite some time. How does one frame their really basic, maybe yeast-oriented molecular mechanism work as a health disparities problem? And one thing, I mean, I'd love to hear from all of you about this, and we would collectively, so we can connect you to Evelyn's office, to to develop these ideas further. But one thing that occurred to me during Evelyn, Dr. Breuer's talk was that if underrepresented populations do not get in to see their doctor until a late stage of cancer, then a certain kind of genomic aberrations are going to be overrepresented in that population, and those might be dealt with through different mechanisms that you guys study and are developing targeted therapeutics for. And on top of that, as many of you have already explored, some of the pathways you're studying are differentially expressed or function differentially, or the protein products are differentially either implicated through GWAS studies or other mechanisms are different in different ethnic backgrounds. So for example, I noticed that Dr. Cooper and Jyoti also talked about U2OS cells in their work, and those are from a single Caucasian female. So being aware of that and really addressing whether your research project is using model systems which can represent the vast diversity of people that we see in clinics is important. So just to quickly end on this announcement that we wanted to point out, this is both for R21s and for R01s. There are upcoming dates, particularly in spring, for which this this type of encouraging basic molecular science scientists to have their work focus on mechanisms that may differ in different populations is supported by the NCI and really encouraged through funding mechanisms like this. And if you want any further information of additional mechanisms, then please talk to Tintin, Craig, or I about it. So really, that's the only thing I wanted to point out today. And so with that, I guess we can end almost on time. Anything further from the organizers?
No.
If I could just add real quick, the beauty of these RFAs is they're not asking you to change your scientific question at all, right? It's the same scientific questions you're asking right now, just in the context of sort of taking into consideration some of these differences in genetic backgrounds. So it's a sort of a timely opportunity.
Yeah, and let us know if you need any help. We're trying to develop these resources right now to help people figure out if their pathway is differentially regulated, affected in different populations.
Patricia?